Anyone here recognize where this is? I think that's the Golden Gate Bridge back there. Just thought, a lot of places in the country or in the world, I show this picture, like, where is that? Well, it's just down the road. So I'm going to look at microplastics and along with everything else. I have a somewhat different approach. I also do ecological risk assessment, and I don't do it like EPA does. Because, my daughter could do multiplication and division like third or fourth grade, so we can do better than that now. And there are a lot of people that have helped. Suzanne and that whole group at OSU, they did dose response curves, really wonderful ones. Stacy and everybody. So this is a big effort, and part of this is, Sean, are you here? Yeah, part of this is paid by you because that effort is what we use to get all the seed and serve data in shape to use. So thank you for that as well. This is what we have to do if you have an NSF grant. You have to say NSF paid for it. And maybe in a couple of weeks, months, maybe they won't be there anymore. It's been very bizarre lately. So NSF funded this. Oregon State, it was a consortium. And then Ruth Sofield, at my lab at Western, we also see dose response curves from her when it comes to the leachates. This is how I think of the world. It's a little different. I think my wife may have taken this picture, and I'm doing the flying. This is how I think of the world, not as a single simple stressor. That place right here at the bottom, that's actually a national marine reserve, estuarine reserve. You'd think that'd be fairly protected, but you see the tankers up there going into the refineries? Pretty cool. Anacortes is a harbor. Lots of boat and shipping coming through there. And then this is the mouth of the Skagit River, which has one of the most productive agricultural areas in the state of Washington. So there's no other runoff into this estuarine area, right? So this is my world. And maybe I'm biased because I go through this all the time, but having multiple stressors, mixtures and so forth is just how it is. Which means if you just look at one chemical or one stressor, it's hard to differentiate what's happening there, and that's why we've had a certain approach to risk assessment. And I had to put Wayne's World because I was teaching and the movies were coming out, and my students like to remind me of that. We have very specific questions. How do we calculate the relative contribution of all these stressors, the impacts and so forth we may see to things you care about in the environment? In this case, we're going to really be focusing on the anchovy, northern anchovy, and I'll tell you why we're not doing Chinook. And we've done other kinds of things and reports you can get, but other kinds of water quality from parts of it. How can we provide environmental managers a risk table? This is, if you care about these species, these are the kinds of things that are going to affect it. So you can start making decisions. And can we aid in the process of helping people make those conditions to actually making a management aspect? Well, if you just try to manage stressor by stressor, you'll fail because they're all there together, and you'll fail and you will understand why perhaps that happened. And you can tell EPA regulates chemical by chemical for the most part. I've been many of those committees over the years, and I go, well, that's probably not going to help here. And microplastics. Microplastics are now having the data that we need, my kind of people need, in order to start doing these kinds of probabilistic risk estimations. And thanks to Stacy and you for helping with that. I don't want to talk very much about Bayesian networks because I'm sick and tired of it. If you want to know more about it, there's some papers you can read. The context is in the overall research programs we've had, and we've covered lots of things I'll talk about in just a second. And we have microplastics, mercury, pesticides, leachates, risk to fish endpoints. What's the probability of an impact to a valued resource? How many of you guys gamble? Play the lottery? Anybody here play the lottery? Usually I get a lot of hands. I go, oh yeah, so you play the lottery, so you don't understand probability. They will win. And what we do is we deal with probability all the time. It's just hard to see innately. Over the last many years now, we've done all kinds of other kinds of stressors. Ones that are very well understood, like mercury, carbamates, organophosphates, classic things, water quality, climate change. If you want one that's all probabilistic and really hard, climate change is challenging. PCBs, one of my favorite, and invasive species. So some of these are chemicals and some are not. We've also done risk assessment for gene drives, which are mostly hypothetical, but they're becoming not hypothetical to control things like malaria. We've also, our latest paper is on the Mackenzie River up in Canada, not the little one in Oregon, but the big one, which is, that watershed's about the same size as California, absolutely, and it was mercury, very large scale. And then Puget Sound and the usual places. Microplastics in the context, it's a developing field. And in the 5 or 6 years I've been working with it, it's developed a lot. But now we're starting to get some of the answers we need to actually start doing about risk. Thinking about mode of action and so forth, the kinds of things we have for other kinds of chemicals. And the more we get that, the easier we'll be able to do the risk in comparison to the others, and especially exposure response. And I'm going to keep saying that until I'm done. It's just, come on, this is math. We've been able to do it for years. Which also means that most of the papers you see with an EC50, I don't know what that's supposed to mean. And I've been doing this for a long time. It's not always transferable. San Francisco Bay, that's easy for us to drive to it. And I'm not going to cover the rest of the estuarine area around here because there's just not time. And we're going to look at Chinook salmon and northern anchovy. Why Chinook salmon? Because you have to. And northern anchovy is the endpoint that we're going to concentrate on the most because of the data sets. We're looking at risk of microplastics compared to mercury and bifenthrin. Why we really understand those very well. And there's lots of information, there's great sampling, and so on. This is my diagram of Bayesian networks. I just am tired of it. What it is a very clear way of dealing with conditional probability. The boxes are nodes, and we talk about interactions. You know, when you have an equation like F equals ma, you'll put one of those variables in there, and that tells you how to transform one thing into another. We use conditional probability tables. My background's in genetics. If you're into the genetics, conditional probability is very, very nice. That's how we do it. We also document things extensively. Please write more notes, because this is how we decide, and we tell you how we decided to give things certain values, to say they're not important, so on and so forth. So everything that we do is documented in these tables about the nodes. Also in the software we use, it's very explicit about how things were done. It's called Netica. It's used extensively by people all over the world, and there are other software packages too. The reason we use Netica is that so many other people doing environmental assessment have used it, and we try to learn from what their mistakes were as well, and they have really good examples. And it's been used for forestry, the kinds of things that we do in molecular level, and for medical diagnoses. Okay. If you want to know the background, just look at the papers, and there are 2 reviews on here. I assume that the slides are available, so there's the references. We think about the world in a little different way. I go from sources where chemicals or other stressors come from. We make a list. We want to know where it is. We use GIS maps all the time. Spatial relationships are enormously important. We have effects. We describe those effects with dose response. That way we get the entire curve and not a point on the curve. I've never heard of a curve being defined by a point. Have you guys? But that's how we report toxicology all the time. No, we were going to describe that as much as possible because we will use the entire curve. So we're going to use the entire exposure response, and most people just look at the EC50s. We already heard that. EC50s are nice, but all it takes is a small change in survivorship of the adults at a certain age, and you have a population collapse. Here is some information that's not been reported here, but it's in the theses of 2 of Ruth's graduate students, and these are leachates. If you want to see beautiful dose response curves, look at these curves and the points on them. So we have that leachate probably better defined in this particular fashion for the leachates. Also, the Brander and Harper labs have dose response curves and did the experiments so we could have the dose response curves. Anyone doing toxicity work with microplastics, just call me, email me, and I'll give you how to do those kinds of experiments. Or people still working, I'm kind of retired, so you can ask the people that's still working for that. This is what we end up with. This is the tables, the boxes at the end of the particular model. It doesn't show up, okay. These are the nodes that are important here. This is the Chinook salmon that are going to be affected. We use catch per unit effort. Has anyone ever used an organism called catch per unit effort? You know about these things. But the catch per unit effort is like counting the population size, right? It's population size. That's what we're using here. We're not using population models like we've done in some of the work. And here we have outmigrating Chinook. Some of these other arrows are coming in with all the effects, and the anchovy. And you can see these are distributions. And the distributions are different kinds, sizes of the fish population. Here, this is out-migrating Chinook. See the Chinook that are affected? You can't see hardly any difference in those boxes, can you? The Chinook are now at such a low level, I'm not going to talk about it anymore. Once a population is essentially extinct, you can only make it more extincter. So it's not going to be very responsive. However, the northern anchovy, there is enough distribution into it, there are enough fish at different age, at different sizes and so forth, that you can actually go from here and add the chemicals, and you see that the lower number of CPUs are more probabilistic, have a higher probability. So that's how you see the effect. The risk of being at those lower populations has increased. That's how we do that. And notice these are all distributions. So I'm not going to really talk about the Chinook. It's sad. And the anchovy is where the signal is. Tire wear particles, we've already done some work with that with just the particles. Again, it doesn't change much with the Chinook, and it changes just a little bit with the northern anchovy. And these are particles, not 6-PBDQ, just the particles. And that paper hopefully will come out one day. This is the model we use, and you're going, oh my God, I can't see it. And I don't have time to explain it, but it's like all the other ones we've done. And I'm more than happy to explain it, and it runs just fine. But the important part for you guys is going to be here at the end, and then we've described fiber leachates, mercury bifenthrin, and the the tire particle leachate in the San Francisco Bay. And we split it up into the 4 regions that were in that map, so it's comparative in that way. And this is what it looks like. For the red, green, and fiber leachate, and it's going into this one. This is the anchovy. And also from this side, the pathway is coming the mercury bifenthrin and TP leachate. So we're doing the fibers here and the other kind of material of the pathways on the previous model. And over here is the anchovy before. And look at the distribution change in the northern anchovy. The distribution of the CPU. In both species shows a decrease when the stressors are combined, but the Chinook not so much. It's just hard to see. But it's really the northern anchovy where you see the difference. What happens is the probability of being a lower CPUE is higher than it was before, and it's how we can do risk. It's all about probabilities. And this is just blowing it up. And this is just the sensitivity to findings. Now we know that there's a change. What we're going to do is use something very similar in conception to a non-parametric correlation analysis that if you've ever done Monte Carlo, it tells you which of the variables are really driving it. This is what you're doing for a mutual information case because what we're using are not continuous variables, but discrete ones. So we go through and we did this analysis, and we'll see what contributed to this change in the anchovy population. Oh yeah, you can't read the small box, but that's ding. But the key ones are the total anchovy population. Of course, that's going to be very predictive of what the future one's going to be. Total mercury and bifenthrin. We know those are very toxic, and actually they're being managed in this region anyway. Mercury toxicity, well, of course, that's the dose response curve. Total fiber leachate toxicity, it's the leachate that we have from that. And then again, the mercury concentration. So where do the microplastics show up? Well, pretty much in the total fiber toxicity aspect of it. We had nice dose response curves. Also, we have the data to demonstrate on. Not saying there aren't data for the other things, but not quite as clear. And also, there's some other reasons which have nothing to do with intrinsic properties of the microplastics or these other chemicals. Mercury and bifenthrin have been sampled many more times, over what, 40 years in the Delta and in this region? What kind of data sets do you think we have for those? We have extensive data sets. And remember, a lot of times you have extreme cases that produce the toxicity. But in order to see that entire distribution, you have to sample enough times. How many times, Ezra, how many times have we sampled microplastics compared to these chemicals? Not as much. So what's happened, if you go out 3 times, you're going to see the tails. Unlikely. Where these other chemicals, we have the tails well understood. Also, it's not clear yet from the data sets I've seen how much sampling you have to do to be sure to see those tails. That should be the question that you ask. How often do I have to go out and sample to get in a complete distribution? For bifenthrin and mercury and so many other chemicals, we have years of history for that. We don't really have that for the microplastics anywhere that I've seen so far. And there may be a place, because I do other things too, but I haven't seen that. Suzanne, you probably have a much better answer, and you're shaking your head that way, okay? Bifenthrin and other pesticides are designed to be pesticides. They're toxic. Methylmercury is one of the earliest, Minamata disease and so forth. It's one of the earliest cases of having a clear exposure response causing nervous system damage. So we understand that very well. My first job coming out of graduate school really was working for the Army on chemical warfare agents. Very small differences in those structures make a complete difference in their toxicity. One's a pesticide, insecticide, the other is a chemical warfare agent. So we know that very small differences can have huge differences in toxicity. For the plastics and leachates, the toxicity is not designed in, but it is in many ways a byproduct of what they're intended for and their use, which also makes, there's going to be many things that are unexpected. Why is this chemical doing this? Why is this anti-ozone agent now being biotransformed into something that's highly toxic? That was completely unintended. And also we're so naive about the world that we couldn't predict it. So I think we treat the toxicity investigations in a manner more similar to that than we do for they were like for pesticide registrations. Let's be more careful about it. So I'm saying something do we need to think of this as something comes under FIFRA? Are using the experience from FIFRA to apply our reasoning to this and do the risk assessment? Absolutely. We have so little knowledge that we need to really understand it. Also, when we first started doing risk assessment back in the 1980s and early, most of the chemicals we were looking at actually worked by narcosis. Narcosis is great. We understand that. These chemicals, we don't really understand what their modes of action are so well. And if you want to use the species sensitivity distribution, which was also invented in the 1980s, that's fine with me, but don't have it as the basis for your decision. Because part of it, and we have a lecture on this, is somewhat an artificial construct from people doing things with similar modes of action back in the 1980s. It's widely used. When I call it a piece and making a decision that's widely used, that's an administrative and regulatory requirement, not necessarily a technical one. And this is my best slide. The same area I just showed, I'm a little bit further away from it, and it is a beautiful place. But remember all the stuff that's going on over by where the sun's going down. You can see the refinery, see the steam coming out of there. Beautiful area, isn't it? And think of all the stressors. Thank you very much.
Thank you, Dr. Landis.
And thanks for not snoring. I know it's right after lunch.
I think we have time for 1-2 quick questions. Yes.
Sorry, I thought just update everybody that also if Tricia Lee's still here, she was also
I know.
instrumental in funding.
Where'd she go? Oh, there you are.
So
Okay.
Delta Science Program. I
Yes,
just don't wanna
thank
take
you
all
for
the
your
credit.
support. Yeah.
Any questions? Other questions?
You know, if I was teaching, I'd start asking you questions. So has anyone here actually thought about the process of risk assessment when they're designing their microplastic studies? I see you nodding your head a little bit, but I know, yeah. Yeah. So how do you incorporate that into your into your study? Understanding what we need for risk assessment.
I will say that in my early career, I was not designing the rigs. I was thinking about hypothesis testing. But
Yeah.
I feel like in the microplastics world, a lot of papers have been published on, like, here are the things you should do in order for it to be useful to a risk assessment.
Right.
And I feel like the biggest thing I see, you can tell me if I'm wrong, is multiple concentrations. So a certain amount of concentrations that you have within your treatments, and then a certain amount of replicants
then
within each of those.
you
'Cause
can
then
describe
you
it.
can make a dose response curve.
Yes, you can describe a curve. How simple, right? And the calculation is now trivial. When I first started, it was hard. You had to write the program, and now you can just look up R code and download it from the DRC sites.
Can I ask
Yeah.
a question since I have this microphone in my hand?
Absolutely.
So you made a comment about SSDs.
About what?
Species sensitive
SSDs,
distribution.
species sensitive distribution.
And in both Canada and the United States, they're sort of seen as the gold standard for risk assessment.
Yeah, so it's a gold standard of 1985.
for sure for making decisions.
That's right.
So what is your recommendation? How do you change an old gold standard to something you think is better?
So that's really hard. And it's become so ingrained that people think that's actually risk assessment. I go to a different meeting other than CTAC. I also go to Society for Risk Analysis. Very different approach. And you write papers and you explain what the issues are. You understand that in some of the SSDs, I just looked at one not too long ago. I was on an EPA panel. Like Musk, I was a special government employee. Very interesting. And I looked into the SSD they had. They had data for 4 species. They extrapolated the other 25. How much data did they have there? 4? And the rest was extrapolated. Understand why it's done. But then what happens to the uncertainty? How do you detail that? Can you write a box about how you did each thing and have confidence in it? And the answer is that's really hard to do. Also, those SSDs many of them, not all of them, some actually do have confidence intervals. But remember, that's a point for an LC50. Is it LC50 a data point? I love it. You're shaking your head no. Of course it's not data. It's a compression of all that information that's in that curve into one point. What happened to all the variability and the uncertainty with that measurement? It's gone. And that's what we're basing decisions on. And so you can tell I've practiced this before, because these are the issues. Yeah. Now, if it's something mandated by regulation, go ahead and plug it in, and we can calculate what that would be from this, because it's actually trivial to do. It's not really alive, and hopefully they have the curve. So it's an easy thing to do. That's not really what I say when I've had a beer or two. I just say, just get rid of it. Yeah, yeah. Yeah. Also, when I fly airplanes, we use GPS now. We don't use just time and direction and compasses and things like that. We use GPS. Why not change over? Yeah. Thanks for asking that question.
All right. Thank you again, Dr. Landis. I think we move on to the next talk. And you're on the panel. So, second speaker in session 2 is Dr. Ezra Miller. They're senior scientist at the San Francisco Estuary Institute, and they're presenting their talk on monitoring microplastics to inform management in San Francisco Bay. Thank you for being here today.
Okay. Yes, I am not this tall. Cool. So my talk is going to be a little bit different because I'm not going to talk that much about data and methods and specific studies. It's more a kind of long-term look at what we have been doing in our area around microplastics over the last more than a decade. So before I get started on that, I just wanted to tell you where I'm coming from. For those of you who are not familiar with SFEI, we are a nonprofit scientific organization. We do science to inform management in the San Francisco Bay Area, but also in California more widely, and even more widely than that, nationally and globally. I'm part of our Clean Water program, which is focused on doing both cutting-edge research and more routine environmental monitoring for a variety of different types of contaminants in water and other matrices. And then we also have a big emphasis on communicating our research to a wide audience so that it can be useful. As an example, a long time ago, before I even joined SFEI, we did a really small kind of pilot microplastics monitoring project. And by today's standards of microplastics work, it's terrible, but at the time it was good. And we presented this at, as a poster at the State of the Estuary Conference, which is just a little local conference. And it got reported on in the news, the Mercury News, and it was actually a front page news article. And the time that it happened to be right after the legislature had passed a bill, the microbead ban bill. If you remember way back when we used to put plastic beads in all of our personal care soaps and stuff. And the governor was debating whether or not to sign it and was getting lobbied pretty heavily to not sign it. And then he read this article and he signed the bill. And his staffers told us that this is what made him decide to sign the bill. And that was really significant because it was not the first state bill to ban these microbeads, but it was the most strict. So all the other states had various exceptions. And the federal bill followed the California example, and that is often the case. And so now we no longer have plastics in our soaps, which is great. So following the amazing success of that little tiny pilot study, we embarked on a much larger study, which you've actually heard about in at least 3 of the prior presentations, where we monitored all kinds of matrices in the bay. And importantly, we also thought about the pollution pathways. So treated wastewater effluent and urban stormwater runoff, how the microplastics were getting into the bay in the first place. And I want to be very clear that in California, these are 2 separate pathways. There are only 2 wastewater treatment plants in all of California that are combined sewer systems, which means they treat both stormwater and wastewater, downtown Sacramento and downtown San Francisco. Everywhere else, they're separate, which means that most of our stormwater runoff is going into our surface waters untreated. And that is something that I feel like the microplastics world still hasn't really wrapped its head around, because a lot of the research coming out of places like Europe, where they have combined systems, they don't think about it in the same way because their water treatment systems work differently there. As was surprising at the time, but when you think about it, is not particularly surprising, we saw much higher levels of microplastics in stormwater compared to treated wastewater. And when you calculate out loadings by looking at the amount of water that is going into the bay from wastewater versus stormwater, there's a lot more load coming from stormwater. Wastewater is important, but stormwater is so important. And this has really shifted our paradigm of how we're thinking about microplastics in our research program. Yeah, urban stormwater, really important. And not only that, but the majority of microplastics in urban stormwater runoff are tire wear particles and fibers, which you've also heard a lot about already. This is so tiny. Sorry, I'm like, I want to adjust this. Okay, so once again, this made the news. These are bigger news sources that more people read than the Mercury News. You can see 2 front page articles about microplastics in San Francisco Bay. So this project, I think, is still one of the most comprehensive monitoring efforts for microplastics in the world. And it really, we're trying to change the paradigm. Think about the stormwater, think about the tires. Jackie talked about how much tire particle mass is going into the bay every year. It's pretty impressively high. But we don't just want to be in the news. We also want to work towards solutions. While we were doing this project, the California legislature really got into microplastics. And they passed a few bills that were maybe even jumping the gun on the science a little bit, but that have helped drive microplastics work in California. The first is one thinking about microplastics in drinking water, which is now starting to be tested across the state. And then the one that really affects us at SFEI is this statewide microplastics strategy requirement for the Ocean Protection Council to start to think about microplastics and how we handle microplastics as a pollutant more long term. And I mentioned the drinking water regulations because that led to something really important, which somehow, this is a microplastic session, but we haven't really defined microplastics yet. This definition, which Scott Coffin over there authored, was, I think, the first legal definition of microplastics, regulatory definition of microplastics. And even though it's specific to drinking water, it's been kind of used for all kinds of other matrices. So this is really what we're thinking about when we're thinking about microplastics. So some people might decide, oh, tire wear particles don't count, but we would include them as part of it. I also want to mention, I think Scott's going to talk more about this in his talk, but that bill also led to trying to think more about risk, and several of the folks who have talked or are going to talk were involved in this effort to start to develop thresholds for Wayne's dislike of comparing a concentration and a threshold to start to think about risk of microplastics. And Scott used our data to start to think about how those thresholds compare to data in California. And I'm bringing up this paper not to necessarily be like, oh, it's such a concern in California, but to say that this is what the California Water Boards used in their integrated report, which is the report that they summarize all of the water bodies in the state for whether they're impacted or not. So it's really important that this made it in there because the regulators are starting to think about, do we consider a water body impacted because of microplastics? We were at the same time thinking about how we can deal with the microplastics. They're in the bay. Well, where are they coming from? How are they getting there? So we chose 4 categories of microplastics: tire wear particles, fibers, specifically from cigarette butts versus fibers from other materials, and then other like fragments from single-use plastic type foodware, et cetera. And we came up with these conceptual models for each of them, which outline all of the different potential sources to the environment and how the particles move from source through the environment and end up in the bay. And this is to help us figure out where we can manage these microplastics. And so I'm showing the one for fibers, but there are conceptual models for all of them. I also want to mention, this has been touched upon, but just to be really explicit about it. So we think about microplastic particles, and we think about chemical contaminants, but really, those two things are one thing often. So it's particles and the chemicals they contain together. We have to think about the chemical ingredients of the particles and how they leach out or not, in addition to just the particles themselves, for really truly understanding risk. And I think that 6-PPD quinone is a really great example of this, because we understand that 6-PPD quinone is really toxic to coho salmon. Jackie talked about that earlier and showed the same graph. What we don't quite understand yet is like how much 6-PPD quinone is leaching out of tire particles and for how long and how weathered the tire particles are, et cetera. We see in San Francisco Bay, this is our data, that the concentrations of 6-PPD quinone in our urban creeks, which are primarily stormwater runoff when it's storming, are quite high, concerningly so. But we don't know how this relates to the microplastics, the tire wear particles that are coming off into the environment. I wanted to mention this also because the Department of Toxic Substances Control's Safer Consumer Products program was actually the first agency in the world to regulate 6-PPD quinone in tires, and that's a big deal. They used our data to make the determination that they could regulate this. And so they have this 4-step process for promoting safer product design by evaluating chemicals within consumer products that have the potential to harm humans or the environment. And then they have the authority to require manufacturers to evaluate safer alternatives and enforce regulatory action. And so the 6-PPD quinone is now in the compliance and enforcement kind of state, I guess, yeah. It's in the manufacturer evaluation of alternatives state, but DTSC is also proposing to add microplastics to their candidate chemicals list, which is would be a really big deal because it would allow them to regulate products that contain or have the potential to release microplastics. And they're working on several products right now that are within that kind of category, things like paints and artificial turf. So there's a lot of possibility here in terms of dealing with microplastics at a source control level. But of course, we don't have management just source control. There's a whole suite of management options, everything from preventative options, the source control options, all the way down to remediation options. And I really want to stress here that something that we often forget about is that as you move away from source control and towards remediation, the societal cost of doing it increases. So it may be less expensive for a specific organization or place, but it overall is more expensive and overall is less effective. Source control is the most effective way to deal with pollution. And this isn't just one entity's problem to deal with. Like, there's lots of different actors that can be involved and really important in making this whole suite of different management actions happen. So I've been showing the suite of management actions for tire wear particles, but here are the actual different management actions that we identified as possibilities to help with the tire wear pollution problem. And we did the same kind of management options framework for all 4 of those categories. And the Ocean Protection Council is now thinking about all of these different actions and which ones to fund more research into, et cetera. So I also want to take a moment to talk about some of our ongoing work in addition to stuff that has already been published. Starting with something that I guess is also already published, but we periodically think about our overall management approach or monitoring approach and how it relates to management. And we last year released this update to our strategy for microplastics in San Francisco Bay, and it includes identifying our key management questions and also the data gaps that we think are most important for us to be able to answer. There are other data gaps, obviously, but we don't have the expertise to answer them. But we could maybe address some of these. And we're working with a lot of experts around the world, including Chelsea. We don't have our own lab space, so really all our analytical partners are like key to our success. So shout out to all of them. So one of the projects that we're currently working on is actually on the remediation end of things, thinking about how microplastics and many, many other types of roadway contaminants are removed by bioretention rain gardens. And this is looking at several different bioretention rain gardens in the city of San Francisco, funded by the EPA. This is just preliminary work, but we do see reductions in tire wear particles. This is actually as measured by tire marker chemicals, so tire chemicals, particles all together. Each color is a different rain garden, and you can see they vary quite a bit in both what's flowing into them because of where they are in the city, but then also how effective they are at removing. We also took soil samples, and we saw lots of different types of microplastics, especially fibers. So here's a picture of some of them. And then here's some other examples. It's not just fibers. There's road paint and polyethylene fragments of things, polypropylene fragments. So while fibers are dominant along with tire wear particles, we do see these other types of microplastics as well. The other study that we're working on right now is thinking about dryers. So Suzanne mentioned that washers are important, but if you think about the total load of stormwater compared to wastewater, well, if there's fibers out on the landscape and it rains and all those fibers get washed into the stormwater, that's going to be a really important thing to think about. And we think one of the main sources of all those fibers out on the landscape is dryers, because lots and lots of people have houses that have a dryer vent that look kind of like this. You can see it with your naked eye, all those little fibers. So we have been collecting dryer vent samples from households. These are preliminary, so it's small right now in terms of our sample size, but we're growing it. Households ranged in size from 1 to 4 people with a huge range of ages. And then a whole bunch of different types of things that they were washing and drying. Daily clothing, sportswear, bedding, towels, pet stuff. And then you get a ball of lint at the end. Most of it was not plastic, but about 14% of the dryer vent that would just go out into the environment is plastic. And if you start to do some like back of the envelope, very gross calculations, you can start to say like, oh, actually probably about 3 grams of plastic is vented into the environment per house per year. That seems bad. If you gross estimate this out even more, so I live in the East Bay, I live in El Cerrito, and I have a 3-person household, my spouse, my child, and myself. And the East Bay, about 4% of households are 3-person households. And all of us together, just the 3-person households in the East Bay, are venting out 1 plastic grizzly bear's worth of plastic every year, which is quite a lot. So if you extrapolate that out larger to all the different size households, et cetera, you can start to see, oh, maybe this is a really significant source of plastic to the environment. If you want to help us do this research, you could take this survey. So this is a laundry habits survey. So one of the big assumptions that we're making in those calculations is how often people are doing laundry, drying their clothes. And so if we have more data of more people and how often they do laundry, that could help us be a little bit more sure about some of these extrapolations. So I would really appreciate everyone taking this survey. Thank you. Give you another moment to get the QR code.
think you can leave it up while we have questions. Thank you,
Oh, wait,
Dr.
I have
Miller.
one more thing. I have one more thing. I just also want to mention real quickly, we are also working on a California statewide plan for monitoring plastics, both macroplastics and microplastics together, and thinking about how they're connected. And this is coming at some point in the next year or so for public comment, so stay tuned. And we have so many funders and collaborators, and if you have questions, talk to me after. I know I went long, sorry. Or visit our website. A lot of this work is published and available. Yeah, now I'm done.
Thank you, Essa. You can put up the QR if you want while we take questions.
Hi, if I have time, I have two questions. One on the plastic data that monitoring that you've been able to get, the microplastic data or microparticle data. Have you been able to do any modeling to see if you can predict the data that you're getting? Like try to see what is affecting any kind of other factors, time of year, weather, wet year versus dry year, those kind of things. I've seen some work on Washington. Apparently they're using traffic data to predict tire wear particles in the environment. So have you looked into doing that?
So we haven't done any of that yet. It's something that we have thought about. Our data set is actually pretty small, even though it's the most comprehensive data. It's not that much data. This is something Wayne brought up too. Like compared to many other more historic contaminants, like we have 50 years of mercury monitoring data, right? We have just like the 2016 to 2019, just a few samples. It's not really enough to do that kind of modeling yet.
Okay. And then the other thing I was wondering, I've asked people about rain gardens before. The maintenance of those rain gardens, eventually they're going to be full of microplastics.
Yep.
I have not heard of anybody actually managing rain gardens to remove the microplastics.
That's a fair and reasonable concern. I agree. I don't know what's going to happen. I mean, this is a problem with rain gardens, not just for microplastics, but for lots of different contaminants, right? Like they're designed to remove a whole suite of different stormwater contaminants, and so they're full of metals and PCBs and everything else. And so dealing with that at some point in the future will be something to worry about. But rain gardens are not the solution that some people think they are. They're very expensive to put in, they're difficult to maintain. They are part of a suite of solutions, and they have their own set of issues that will need to be resolved.
Any other?
I have a question that's kind of related to that. I think when you were talking about combined sewer systems, you were sort of talking about them like they were a good thing. And I'm really used to this, I'm used to this conversation in Toronto that they're a bad thing because they're old and they don't fit our system. And so when we get big rains, they just release it all.
Yeah.
And then we get wet wipes and all kinds
This
of other
is-
things. So in Europe, do they also have these problems? Like, okay, so you're not advocating for
I'm not
combined-
advocating
Yeah.
for combined sewer systems. I'm just saying this is something that we often forget to think about.
Yeah.
And especially I hear all the time, oh, this is all just a wastewater problem. And I'm like, no, we have the data to show this is, wastewater is a concern, but stormwater is a much larger concern, for me at least.
When you were sampling stormwater in San Francisco during the study, were wastewater treatment plants releasing or not wastewater treatment plants, but were they letting it all out, the sewage too, because of the rain or
So
no?
San Francisco just has downtown San Francisco is the only wastewater treatment plant that is combined, and it releases into the ocean. So it's irrelevant to us. Yeah.
Thank you.
And at the Sacramento wastewater treatment plant, you have storage basins on the property to collect rains during temporary heavy storms that are then treated as the storms diminish. And the downtown area also has water reservoirs that fill with that storm water. If we're gonna have a 2-week downpour, we might get into an issue where that combined system will not be sent to the treatment plant. But for the most part, we haven't had that happen in a long time.
Was there any other question here? Oh, wait. Maybe the last one.
So we couldn't have done a risk assessment without your data set. And to me, it's like the first time anyone ever had a plan to put it together. The biggest frustration is we know how long that was done ago.
Yeah.
And if everyone's so concerned about microplastics, why the hell aren't they out there in the world measuring what they are? This is so different. PFAS, we're measuring that. All the classics are being measured all the time. But we, in so many ways, the uncertainty is less with those because we understand them so much better. So we have something that everyone says is so important, both in academia and so forth. And occasionally you have someone in the legislature said, oh yeah. And why aren't we measuring it in a routine way?
It's too expensive.
It's not.
It's all money.
Yeah, I know. Yeah, so I'm well aware. I used to work for DOD. We actually had real money there. You know, it's a completely different thing. But it will answer the question. So here we have something that's clear. It's like, why are we building another big telescope to put in space? Because now we can see all the galaxies like forever. Why don't we spend a fraction of that to start measuring what these materials are, both in plastic itself and the materials that come from them? This is don't have to invent a lot of new technology. It's just a lot of work to do. If you're serious about answering the question, you have to have the data, or else you're just whistling past the graveyard, I think. But I'm retired now, so I can get away with stuff.
Yeah, I will just say that the data that everyone is using is from 2019. That was pre-pandemic. Things may have shifted. We don't have $1 million to do a repeat of that study. And so it's not as high on our priority list of some of the other data gaps that we can answer. But no, that's what I'm saying though, is that this is a challenge just in general, is how expensive and time consuming and so much work to extract microplastics from the environment. It's not even necessarily the microplastics that we're most concerned about. And so, yeah, it's still a big open-ended question issue.
Why not? Why don't we just start building a plan? We have your guys' stuff. We know a lot more than we did in 2019. To actually go through here, these questions that need to be answered. How do we sample the environment to do that? I have not heard that. Maybe Scott will have the answer here in just a second. The next speaker will have it. But I've not seen this in Europe either. I've not seen this in the papers that are out. God knows, people have made entire academic careers from these microplastics. Until I see that happening.
Maybe that's a good question that we could keep also for the panel discussion. I mean, it seems like,
Yeah.
no, it's an important question. Thank you, Dr. Miller, for your talk. Yeah, for the sake of time, we'll skip the break. No. Everyone can, of course, like, get coffee in between. And we would like to move forward to session 3, if that's okay. Yeah? Okay. If everyone is ready. Session 3, Dr. Scott Coffin. He was introduced now several times. He's a research scientist at California OEHA. And his talk, oh my gosh, you're killing me. You can read it. You can read it. We're very glad you're here.
And I'm super glad to be here with everybody as well. Nice. So today I'm going to be talking about a comprehensive probabilistic framework for deriving microplastic hazard thresholds and how we developed and applied it to both marine and freshwater ecosystems with what you'll learn is TOMEX and TOMEX 2. So first off, with the necessary disclaimer, I work for the California Office of Environmental Health Hazard Assessment, which is part of the California Environmental Protection Agency, and the views in this presentation don't necessarily reflect those of OEHA or the EPA. So moving forward, something that Ezra covered earlier, Senate Bill 1263, passed in 2018, basically mandates that the state think about doing something about microplastics in lots of different ways, not just monitoring, but also coming up with ways to assess risk and intervene. And in 2022, notably, we developed the statewide microplastic strategy with the Ocean Protection Council. And that policy really sets the framework and the stage for all the research that I'm about to show you. So one of the requirements of that statewide microplastic strategy is that we consider adopting regulations for microplastics for drinking water for humans, as well as for the ecosystem. I'm not going to talk about what we did for humans today. That's a different presentation, different topic, but I do want to talk about the health effects workshop, where we had experts in microplastics from around the world, including several in this room. We've got Ezra and Dr. Chelsea Rockman and Suzanne Brander. And we screened the literature and asked these experts, give us answers on what dose metrics determine toxicity. Is it the particle volumes? Is it the surface area? Also, what particle characteristics are more or less associated with toxicity? How important is size relative to the shape, or does the polymer type matter? What are the adverse effects, and can we use the information to develop a threshold framework that could eventually be used for regulations? And as we learned from other talks today, microplastics are not just particles. We find biological vectors. We find that these are biological vectors for pathogens, as well as, of course, the chemical additives and chemicals that they can absorb in the environment. But for our workshop, we focused primarily on the physical effects of microplastics, as we had a lot of research at the time indicating effects due to those particle-induced toxicities. And for marine organisms, there's 4 main ways that particles can be toxic. Fibers and larger conglomerates can entangle biota. Food dilution is probably the one that we have the most confidence about. This is a very easy one to understand. Basically, plastic doesn't have nutritional value, and it takes up space in an organism's gut and can cause starvation. Internal lacerations can occur as well, and then cellular tissue translocation associated effects, basically really small particles interacting at the cellular level. The main challenge that we found with relating these hazard studies to the environment is that most laboratory studies are done using whatever particles they can get. And most of the time that means fairly simple mixtures of often what we call monodisperse. So this is like one type of polymer, one type of size and shape, usually spheres. They're often pristine. They have sometimes unknown, most of the time unknown additives, even though they are often try to be contaminant-free. And we don't find these types of particles in the environment, really. In the environment, it's every snowflake that you can imagine, ranging in different sizes, shapes, and typically weathered, almost always biofilms and contaminants. So in order to solve this misalignment, there are several methods that have come up based on what's called the ecologically relevant metric approach. So the idea behind this is that the mechanism of toxicity, in this case food dilution or tissue translocation, can be used to relate these different particle shapes and sizes to one another. And for food dilution, we care about the particles that can be ingested by the organism. So if it's larger than the organism's mouth, we don't really consider those particles to be relevant. Similarly, for tissue translocation, particles that are too large to be uptaken by cells or across tissue boundaries, we discard from analysis. And then we take what's exposure metric to be what we consider to be relevant, and then relate that to the environmental effect concentration. So a very simple example here, food dilution. We care about the ingestible sizes, and we are just considering the volume of the particles. And so I'm going to break it down for you. Basically, this is a really simple example. So imagine an effect threshold in the laboratory of 4 particles per liter of polystyrene spheres of a known volume. Now we can estimate how many ingestible environmental particles would fit in that same volume, considering the diversity of particles with a realistic distribution. So it could be 4, it could be 6, it could be It could be 1,000, depending on the size and shape of those particles we find in the environment. And we have to consider the bioaccessibilities in a species-specific way. So different organisms of different sizes are going to be able to ingest plastics of different sizes. Zooplankton can eat particles from a certain range. Fish and Daphnia are going to eat larger particles, and so on and so forth. We also consider the particles in the environment and understand that when you get smaller, you see more of them. There's typically exponentially more microplastics the smaller you look. We don't have ingestible plastic size data for every species in the known world, but we do have a really good model that relates the body length of organisms to the amount of plastic that they've been seen to ingest in the environment. So this is what these transformations look like mathematically. You don't have to understand all of this, but this is kind of what we're doing on the back end. And we applied this to the state of the literature back in 2022. We screened the literature for in vivo aquatic microplastics toxicity studies, and we found that there were 167 with 117 species. We then screened these for a set of quality criteria relating to the applicability for risk assessment and the reliability. And that left us with a very small subset, just 22 studies and 16 species, that then underwent additional scrutiny by experts. Each one of these was aligned, and then we did species sensitivity distribution modeling. And as we've learned earlier today from Wayne's talk, some of the inadequacies with species sensitivity distributions, they are indeed the gold standard used by most regulatory organizations in the world. And this is what they look like in practice, where you have the most sensitive species on the low end, the least sensitive species on the high end. We typically look at the 5th percentile, like the modeled fifth percentile, to protect an ecosystem. We adopted this approach with the consideration that we had a lot of uncertainties with the toxicity studies that we're evaluating. And so we made some decisions in a 4-tiered threshold framework that would allow us to have a set of conservative assumptions versus non-conservative assumptions. So basically on the low end here, we're taking a lot of conservative assumptions in our modeling, and any concentration below this, we would have relatively low concern of being an issue at the time. Whereas on the other end, the source control measures tier here, above this, we would be more concerned about effects likely occurring in an ecosystem. And we did it for both the food dilution and tissue translocation ecologically relevant metrics. Our experts had high confidence in both the framework and the analytical process used to derive these thresholds, but they had low to moderate confidence in the underlying toxicity studies. So of those 22 studies, none of them were perfect, and taken as a whole, there were a lot of hesitations about using even the 22 of the best studies. I'll then talk a little bit about what Ezra mentioned as well, which is how we actually apply these risk thresholds to monitoring data in the Bay. And looks like something that happened with the slides here, but basically what we did was what's called rescaling. And the idea behind this is if you know the shape of a particle, in this case, an iceberg, and you measure the part that you can see, but you know the whole shape of it, then you can estimate what you can't see. So in the case of the San Francisco Bay monitoring, they sampled down to 333 microns with a Mantitrol net, and they characterized the distribution with respect to size. And so with that, we can estimate the particles that they would have seen if they looked all the way down to 1 micron. An additional tricky thing with microplastics analysis is it takes forever to do. Dr. Rockman's poor graduate students that put this together. This is probably what it looked like in the lab. And so they took a subsampling approach to confirm the plastic particles. So they're not characterizing each and every thousands or even millions of microplastics that they found in their samples. We also know that the fibers sampled in this study are not of the most reliable quantification because they can slip through the nets. They're just tricky to work with. And so we adjusted for all of these uncertainties and cleaned up the data in a number of different ways. We did this completely probabilistically, knowing that with each step that we're making these rescalings and alignments involves some level of uncertainty. And so we propagated all of that uncertainty using a Monte Carlo-based approach. And that provided us with what's called correction factors. And you can see the distribution on the bottom right. That's the distribution of correction factors applied to the samples. These numbers are pretty large. So the numbers that they're collecting, the numbers that they're reporting in the samples are quite small relative to what we expect to actually be out there in the water column. And when you relate these concentrations, the rescaled concentrations, to our risk thresholds, that 4-tiered framework, with the uncertainty bounds, it looks like this. So between 25 and 100% of samples in San Francisco Bay, we're exceeding that investigative monitoring threshold, threshold 1. And threshold 4, which is our, oh crap, we have a serious problem here, threshold, we had anywhere between 0% and about 15%. So there's a lot of uncertainty with this assessment, and that's just characterizing the uncertainty on the environmental side, the monitoring side, not considering all of the uncertainty with the thresholds. And so you saw this figure that Ezra showed as well. This is being considered by the State Water Board for additional monitoring under the Clean Water Act. And to my knowledge, this is the first time that microplastics monitoring data has ever made its way into a traditional regulatory framework like the Clean Water Act. Some of these water bodies received a 305B listing, which is not as hard of a criteria as a 303D listing, which would basically recommend a TMDL, or total maximum daily load, be developed. But basically we're saying we don't have enough information to determine that the beneficial use is either supported nor is it not threatened. So looking from this study how we could do better, the missing fiber data introduced the highest uncertainties to the assessment. This is based on a sensitivity analysis that we ran with the Monte Carlo. So the subsampling in the lab had very low uncertainty associated with it. That was not an issue at all, in my opinion. The size rescaling had some significantly more uncertainty, but nothing compared to the corrections we had to do for fibers. One of the ways that we can reduce this uncertainty going forward is by changing our sampling protocols. So using in situ high volume pumps can reduce sampling uncertainties as they can monitor down to smaller sizes. And they can also eliminate a lot of the uncertainties a lot of the self-contamination. Ezra also mentioned this statewide microplastic strategy that the Ocean Protection Council developed. Just to break this down a little bit more about what we're doing with this going forward is this strategy is broken into 2 tracks. The first track is what we're calling no regrets solutions that we can take today, such as pollution prevention, pathway intervention, outreach, and education. And the second track is an iterative approach where we're doing science to inform future action, including more monitoring, advancement of our risk thresholds and applying these risk thresholds, prioritizing sources and pathways for intervention, and then testing out new solutions and repeating the process. So showing this statewide microplastic strategy in action, we are evaluating newer microplastics toxicity studies that were published since that 2022 report. The studies tend to be on the rise as the field gains traction. And we built a way for researchers to not only access that information, but also to upload published toxicity data so that it's in a living database. This is the Toxicity of Microplastics Explorer application. It's a free open source RShiny-based web app that users can go to explore the toxicity data, develop their own SSDs, and also upload published toxicity data. We enlisted anyone that wanted to help to go and mine additional toxicity data from the literature to develop what's called TOMEX2. We were able to get 67 contributors from 14 countries. Dr. Ezra Miller in the room was also instrumental in getting that cooperation and all these volunteers from around the world to help out. And this added 125 new aquatic ecotoxicity studies to the database, bringing our total species count up to 168 and nearly 13,000 toxicity data points. With this new data set, we're now able to split our thresholds into freshwater and marine. We have enough species to do this. And so our total number of species that are being filtered through is more than double. So then we applied the same approach with the alignments, but this time we're taking a novel approach to how we derive the thresholds, taking a fully probabilistic manner in applying the alignments, as well as dealing with the species inter-laboratory validation or variability, excuse me. So instead of just aligning each data point using single numbers, we're doing this with full distributions of actual environmental data using a Monte Carlo-based method. So this is what distributions of particle characteristics looks like in the freshwater versus marine. They're pretty similar for length to weight ratios, but they can be quite different for other parameters like length, mass, and surface area. And then we also take in consideration the distributions with the organisms themselves, such as the body length to mouth size ratio, tissue translocation size limits. And what this looks like is for each individual data point for a particular species exposed in the lab, we get a full distribution of the aligned values. And these can be relatively low coefficient of variability to pretty high coefficient of variability. It really depends on a lot of multivariate factors. And we explored this multidimensionality using sensitivity analysis. We can understand the value, value on the x-axis is the derived threshold with SSD, and then on the y-axis is the nominal value for whatever compartment we're looking at. So in this case, food dilution, when you have a higher volume alpha value, you have a higher threshold, whereas other parameters might not have much of a discernible effect or correlation with the final threshold. So this method is different from others that we've seen in the literature, and I'll break it down a little bit. The original species sensitivity distribution model provides a uncertainty bound based on the model itself, the distribution that's actually fit to the data. A probabilistic species sensitivity distribution has been around for nearly a decade now, and it's been applied to microplastics in 2019. And this considers the variability within the studies, between the studies for an individual species. So this is actually preserving a lot of that inter-laboratory variability in your assessment. And principally, it's not trying to fit some theoretical distribution to the ecosystem. It's actually reflecting the underlying variability in that data. So we took this one step further with the alignment probabilistic method and folding that into how we derive the thresholds with Monte Carlo simulations. And so for, let's see, an HC5 for our tier 3 analysis, we would get a full probabilistic distribution of the thresholds, as you can see here. We iterate this for marine and freshwater. We do it for tissue translocation and food dilution, ecologically relevant metrics. And now we can compare the sensitivity and variability relative to the other methods. So what's shown here on the green is a traditional SSD. We don't know the underlying distribution in that uncertainty. We just get a point value and confidence intervals. With Monte Carlo aligned ERMs, we can get a full distribution, but it's not considering the laboratory variability, whereas this PSSD++ does. It looks a little bit different. So in effect, we have more understanding of what's going on under the hood with all of these complicated modeling steps. And we're using better studies, and we're using more of them. But we also wanted to look forward and see, okay, what additional information do we need? Are we doing things better or worse over time? We assessed the quality of these TOMEX2 studies and found that overall, the applicability for risk assessment is actually going down over time. Studies are not getting better as time goes on in the microplastics field. It is pretty frustrating. And as one would expect, we typically see better studies published in journals that have higher impact factors. However, it's not a one-to-one ratio. Some of the best studies published are in so-called mid-tier journals like Science of the Total Environment, Environmental Science and Technology. Some of these really big journals sometimes publish studies that are not the greatest. So you really got to look towards the underlying study itself and not just judge a book by its cover. And also look to journals that are adopting criteria for publication. So Science of the Total Environment became the first journal to actually have standardized criteria for microplastic studies. If it doesn't meet certain criteria, you can't publish it there. So those standards are really important to moving the field forward. In addition to harmonization of how we report information, as well as performing some targeted studies, and in my opinion, making this as open and easily accessible as possible, which is one thing that the microplastics community has done particularly well, in my opinion. A lot of open source and free resources exist and have been developed by the microplastics community. This is just a really small subset of the over 180 resources that I have accumulated and put onto a website called plastiverse.org. So I encourage you all to check that out. And if you find other resources that could be used by the community, please let me know. So thanks, everyone.
Thank you, Dr. Coffin. Maybe some brief questions.
This is related a little bit on the last slide that you're talking about the quality. So it's discouraging to see how many studies were discarded. So what were the most common reasons, or what are the most common mistakes that people are making?
Great question. So the number one criteria that filtered things out, the bottleneck criteria, was not using at least 3 exposure concentrations. A lot of studies were just using a low and high, or just a single concentration relative to a negative control. And so we can't really do much with that from a risk assessment perspective. Very few studies had full dose response curves. But that wasn't one of our weeding out criteria, because then we wouldn't really have a database.
All the way here.
Great talk. Just a question about the tissue translocation, because that's something we're trying to get a handle on. You showed that distribution, and the tail end was going all the way out to 0.5 millimeter. Just wondering, are there certain taxonomic groups that are out there at the tail end, or does it vary widely? And do you have any theories as to why some species are, we're seeing translocation of larger particles versus smaller ones in others?
Great question. So as part of this analysis, we looked to all the tissue translocation studies that were in our database. I think there were like 50 studies. And we extracted data in a binary way. Most of the studies were reporting it either translocated or it didn't. Very few studies reported what quantity or like what concentration were. And so we did logistic regression. We tried assessing by different factors, like did the species matter, did the polymer type matter. The only relevant factor, or the only statistically significant factor that we found was size. So we could only build a very simple logistic regression model that only considered the size. So these tails, that could be due to studies looking at organisms with different cell types. It could be like organisms that have like gills or like different biologies. It could also be due to false positives with dyes from the particles that are leaking. Sometimes like these like co-spheric dyes the chromophores can leak out when you're looking at it that way.
Right, if there are no other burning questions, there's a burning question, okay.
Can you talk a little bit more about how California's adopted the risk assessment framework? Is that what you said? They actually used it to rank, and will they continue? And will they
Yeah.
put money into monitoring so they can keep using it, to Wayne's point?
So we haven't really adopted the risk assessment framework in a formal fashion, other than the fact that we've applied it to monitoring data for the integrated report. So for that assessment of impairment under the Clean Water Act in San Francisco Bay, that's really the only like official act that we've done with that framework. And the principal reason that we put the water bodies on the 305B list, basically the soft list, instead of the 303D hard list, was due to the thresholds themselves having a lot of uncertainty and uncharacterized uncertainty. So my hope is that once my group finishes publishing this updated framework, that we'll be able to go back to new data or even go back to the same data and make a more formal decision. We'll see though.
All right, maybe additional questions later in the panel discussion. Thank you, Dr. Scott Coffin. Okay, and then we'll move to the last talk of the third session. Dr. Neil Tangri, he's a senior research fellow at UC Berkeley's Goldman School of Public Policy. And his talk is about the need to address plastic production as the primary solution to microplastic pollution and where this conversation stands in the current treaty negotiations. Thank you, Dr. Tangri.
Thank you. Okay. That's a lot of hard acts to follow, so I hope I can keep you all awake. I'm not going to give a research talk. I'm not a microplastic scientist, so this is more on the policy landscape, particularly the international policy landscape, which is where I spend most of my time. So as you just heard, I wear two hats. I'm at the Center for Environmental Public Policy at UC Berkeley. But I also work as the Senior Director of Science and Policy at a global network of environmental justice organizations called Gaia. We have about 1,000 member organizations in 92 countries working to end plastic pollution, working towards zero waste and related issues. And mostly what I want to do here today is encourage all of you as scientists to get involved in policy work. Policy needs to hear a lot more from active scientists. You have a lot to offer, and believe it or not, I know it's going to be a hard sell in this day and age, but policy work can be fun. So I want to encourage you to do that, and so let me dive in. So by way of background, you all know that micro and nanoplastics originate from almost every stage of the plastic life cycle. Leaving aside the extraction of the raw materials, everything from the polymerization, the manufacture, transport, all the way down to final disposal. We call it final disposal, but we still see microplastics in even incinerator ash, right? And so there are macroplastics, which eventually become microplastics, and microplastics emitted really at every stage of this. And that is important because when we talk about policy solutions, the policy solutions tend to be pretty targeted, right? We heard about the microbead ban, which has been one of the most successful and relatively widely adopted policy solutions, and that really takes a very small chunk of this. And so for some of these, we might have solutions. For some of these, we are still very much looking for solutions, and so that creates a very gappy approach. I look at this as a pipeline, right? This is a pipeline of plastic material moving through society, and it's a very leaky pipeline. And so when you have a leaky pipeline, in addition to plugging the leaks, one thing that's very important is that you slow the flow of the material, right? Higher pressure in a pipeline means you're going to get more leaks. If you slow things down, you put less material through, you're also going to reduce the leakages. And so that really brings us very much to the upstream end of things about how do we reduce the production and the consumption of plastic throughout society. And so that's a bit what I'm going to talk about. And in particular, I want to give you a little bit of a history on the international policy process. And apologies to those of you for whom this is old hat. In 2022, we had a meeting of national governments called the United Nations Environment Assembly. It was the 5th such meeting. 125 countries came together and unanimously agreed on Resolution 514, which we refer to as the mandate, because it mandated a new negotiating process that would lead to an international treaty. And this is really a remarkable document. It's quite short, especially for a policy document. It starts with the title. The title is to end plastic pollution, not to address it, not to mitigate it, not to do something about it, to end it. So it sets the standard quite high. It takes a full life cycle approach, and that's really significant because, as you know, this conversation started with like, oh, we have a marine plastic problem, right? We have a sea turtle problem. And now we're like, no, we have a problem all along that whole life cycle that we're talking about. And it talks about every compartment of the Earth system. So it's not only oceanic systems, we're talking about human health, we're talking about really everywhere on planet Earth. It also gives unprecedented recognition to waste pickers. So this is an important part in terms of the environmental justice considerations. We're not looking at this narrowly, right, through this sort of a strictly biological or biochemical lens. We understand that any policy is going to have implications on people, and that there are going to be social justice implications, and the waste pickers in particular are active players in this process. And this art piece by Benjamin von Wong, we walked by twice a day, every day, going in and out of the negotiating sessions, right? It's called Turn Off the Plastic Tap, and it really alludes to the need to get upstream and turn off the flow of material. So that resolution launched what's called the Intergovernmental Negotiating Committee. We've met 6 times over the last 2.5, almost 3 years now, which is a very ambitious agenda to write a new international law. The original plan was to finalize the text at INC 5, which met in Busan in December, and that didn't happen, and you may have heard that that was a failure, right? The negotiating process failed. Well, it hasn't failed yet, because we're meeting again. I'm actually less pessimistic about that. I think that that was an unrealistically ambitious time frame on which to negotiate something that challenging. But I'm also ambitious because we made some real progress, and we made progress in 2 distinct ways. One is we made progress on critical issues, where we're moving towards agreement between countries. Remember, these are national governments negotiating with each other. And also because we had some lines drawn in the sand. We got some clarity from different governments on what are really critical issues. So the substance progress is on things like the financial mechanism, right? We know we've been hearing about the lack of money for research. The lack of money is also a problem for implementation. And so there is a widespread acknowledgement that whatever policy comes out of this, it's going to need money. And where are we going to get the money from? And there are some variety of proposals out there, but there's clearly already in Busan we start to see convergence between governments. There's movement on plastic products. You know, we understand, like, there's a certain amount of products that are unnecessary or highly likely to end up in the environment, and that we can be getting rid of those. There's progress towards non-binding rules on design. So how do we, again, move upstream, redesign things so that we're more conscious in the way that we're using plastic? And on just transition, again, touching on waste pickers, but also indigenous peoples, workers in the fossil fuel and plastics industry, making sure that they come along. I will say microplastics are mentioned only in passing in the text. There are not really specific provisions on microplastics, and I think that that goes to an understanding or a lack of kind of ideas for how are we really going to regulate microplastics per se. The one that's in there is, again, a global plastic bead ban. That's kind of the one possibility. And of course, none of this has been decided. Nothing's decided until it's all decided, as they say. So there is a showdown building because there are some issues on which countries are very far apart. The next meeting is going to be in August in Geneva, although there's lots of side consultations happening this week and last week in Geneva, at the Basel Convention meetings. Governments have been talking a lot. But we have a super majority of countries that want a very ambitious plastics treaty. We had 105 countries standing up in Busan demanding a legally binding cap on plastic production. So think about that. All of the petrochemical industry that's growing at 4 to 5% a year would be faced with phasing down its production. That is a very politically ambitious demand, and it's coming from a majority of countries. 94 countries want controls on chemicals of concern, right? These are the additives, the PFAS, et cetera. But it would also include polymers, right? So some of the particularly problematic polymers like PVC, polyurethane. Some countries are also pushing for a plastic polymer fee. There's less agreement on this one, but this would be a per ton fee that is applied to the production of any plastic polymer and that then could be distributed to go towards solutions. This idea is not as well fleshed out or socialized, so it hasn't moved as far yet, but I think it is one of the more promising ideas to actually answer the question of where's the money going to come from? At the same time, we are facing opposition from a handful of countries who want no controls on production. And these are, unsurprisingly, major oil, gas, and plastic-producing countries. Saudi Arabia, Iran, Russia, and I am concerned that in Geneva we're going to find that the US is going to join that list. And in addition, they're saying we can't possibly impose costs on the petrochemical industry, which kind of blows my mind, but that is the argument that was being made. So how is this, how are these two camps going to find a way forward? It is very risky to make predictions, so please do not quote me on this. Hope maybe something else entirely will happen. But I have sort of 4 scenarios that I can see playing out. One is that the treaty process could just stall. These two camps dig in. Neither one really budges. No progress is made. These negotiations are expensive. And there's always another geopolitical crisis that could overwhelm things. And so we may not get anything out of this. The second is we get a waste management treaty. That would be a really weak treaty. That would be a treaty that's just like, okay, we're going to ignore that entire pipeline that I showed you at the beginning and just deal with stuff right at the end. There's a lot of pressure building on the African countries and the small island states who have been among the most progressive to kind of back down from their demands and just take a bit of money to do better waste management. That would not be a powerful or an effective treaty. The third possibility is that they force a vote. Everything within the UN within the Intergovernmental Negotiating Committee has more or less happened by consensus. But you clearly have a majority of countries who want one thing and a handful of countries who are holding out. And if they decide to force a vote, they could get something through. And if they're not willing to force a vote, then the other thing that a number of countries are talking about is taking this whole process outside the UN and doing what's called a coalition of the willing, where you get together the countries that say, look, we want a treaty, right? The EU wants a treaty, Africa wants a treaty, most of Latin America wants a treaty. Let's have a treaty, and then we'll worry about the naysayers later. So either of these approaches, I think, could end up with an effective and ambitious treaty. The challenge is it will not be a universal treaty, right? So some of the largest polymer producers, including the United States, will be out. But quite frankly, the United States is not going to join any effective and ambitious treaty. So any treaty is going to have to figure out how do we deal with countries who are not parties. And the answer to that is non-party trade mechanisms. In other words, you use your trade to impose restrictions on trade in plastics or plastic products or plastic waste between parties, that is, countries that have signed and ratified the treaty, and non-parties, treaties who are outside. Now, this is consistent with the WTO. Everyone worries about the WTO. The WTO has made very clear that if this serves the environmental purpose of a treaty, it is consistent with the WTO. But it does take, there will be a political cost to imposing those kinds of trade provisions. And a few months ago, I was skeptical that countries were going to be willing to use their trade laws to force through an environmental provision. And I will say one of the things that we have seen with global trade being upended over the last couple of months is that I think there's going to be a lot less unwillingness. I think that there's going to be much more interest on part of a lot of countries to use those tools. So, you know, who knows how this is going to go? Stay tuned. But I do see this is, I think, one of the more promising paths forward in international policy. So I want to briefly mention federal policy. For obvious reasons, I will be very brief. We can't expect much. There are rumors on the Hill of a Save Our Seas 3.0 treaty. Save Our Seas 1.0 and 2.0 were sponsored by Senator Sullivan from Alaska, and it was one of the few pieces of legislation that really moved forward with bipartisan support. Alaskans are very, of any political stripe, are very concerned about their coastline. But, you know, with the change in the political winds, it's not really clear whether that's going to happen or not, or whether it's just going to contain a bunch of gifts to the petrochemical industry. What I do think is going to be important in terms of federal policy is the deregulatory agenda, right? And so this is maybe more of a defensive battle to fight, but we're going to see more and more attacks on EPA. Clearly, already NOAA is being gutted and hollowed out. And so the ability of federal agencies to enforce the laws that do exist and to continue with the rulemaking process is going to be under attack. On a more hopeful note, we have California, and there are some folks here who, I can see you laughing, know much more about this than I, so I'm only going to give a very touch on this very briefly, but I think this is a great place to engage, actually. You know, Scott was just talking about the microplastic strategy here in California, which was adopted in 2022. And the microbeads ban, we heard from Ezra, there is now an expansion of that microbeads ban, AB 832, which is before the Assembly, which would expand the ban to cosmetics and cleaning products, right? So that seems like a logical next step. And the strategy does include a number of really sort of progressive elements, bans on polystyrene food containers, bans on cigarette filters and vapes, all of which are, as you know, major sources of microplastic contamination. There's also an element of the strategy that's about using the state's purchasing power to move away from single-use plastics. So there's a number of things on the solution side, on the action side, as well as a really fairly detailed, as you were hearing from Scott, a really fairly detailed science research agenda. I will say the hot issue right now is SB 54, and this is part of that agenda. This is not specifically focused on microplastics, it's focused on macroplastics, but these, as we all know, become microplastics. A little bit of history. So in 2022, there were enough signatures were raised to put forward a ballot initiative that would have banned polystyrene and create really strong restrictions on SUPs. SB 54 was kind of the compromise to get industry on board. So that ballot initiative was pulled. SB 54 has passed, right? This is law now. And we're in the regulatory rulemaking process, right? It reduces single-use plastic and foodware by 25%. It mandates 100% of packaging and foodware be recyclable, and that 65% of plastic single-use plastic products in foodware actually be recycled, because of course there's a big difference between recyclable and actually recycled, and takes aim at styrofoam as kind of first line of enforcement. So CalRecycle spent 2 years developing regulations on this, and Governor Newsom just pulled them at the last minute. That's not the end of the road, because the law is still the law, and CalRecycle still has to finalize those regulations. And so now they are entering an 8-month period where they are continuing to develop the regulations on SB 54. And this is a time when they actually need to hear from people, right? This is a great time for scientists to stand up and intervene and go on the record. And I have to say, not just with CalRecycle, because CalRecycle is the regulatory industry, they're kind of in the crucible, but talk to your legislators and talk to the governor's office so that they know that there's public pressure here. Industry is pushing very hard here. Industry really wants to gut SB 54 through the regulatory process. They want to bring in chemical recycling. SB 54 is very clear that any process that turns plastic into fuel is not recycling. They want to roll that back and say, well, plastic to fuel is a form of recycling, and then pretty much anything can be recycled, and we're going to call it all recycling if we send it out of state to be pyrolyzed and God only knows what. They're using accounting tricks like free allocation mass balance, which if I try to explain it to you, I think we'll all be on the floor, but I can try. And they're really trying to undo the statutory restrictions here. So this is a big political battle. The plastics industry has been energized by what they see happening at the national level, and so they really want to come after California policy, and that will send, as we heard, California is usually the leader on this. Other states are looking to California. If we manage to get in place good regulations on SB 54, it's going to send a signal to other states. If we back down, that will send a different signal. So how do you get involved? Well, one is, I would encourage you to join this group that Suzanne mentioned earlier, the Scientists Coalition for an Effective Plastics Treaty. This is a self-organized group of scientists who have been engaged with the plastics treaty negotiations. There's more than 400 of us from 70 countries. And we go to the meetings, we meet with government negotiators, we write policy briefs and like short little 2-pagers. And I will just say negotiators are extremely, extremely appreciative. One of the things you have to realize about international negotiations is the US can show up with a delegation of 40 or 50 people. There are 40 federal agencies engaged in this negotiating process. Most countries bring 1 or 2. And they're also covering about 3 or 4 other treaty processes. So their heart is in the right place, but their knowledge can only go so far, right? And they do rely on independent scientists to inform those negotiations. So it is a great place to plug in, and now there's a structure to do it. And Suzanne, are you still on the steering committee? She's too modest to mention it, but she's on the steering committee. And if you, or you can come through me, or you can go to the website. But we would welcome more people involved in that. It's a really important role to play an honest broker in those negotiations. There's a lot of pressure groups. There's a lot of industry. I will be very frank. There's a lot of industry disinformation out there, and having scientists who know the literature and who know the science and can say, actually, here are the facts, is very, very important. And I think that we could use something similar for California. We have a lot of important policy moving forward in California, and I think that I would encourage all of you to be involved in that. And I think we could use a similar structure. So just to sum up, I don't see a lot of, I shouldn't say no policies, there's one policy, but I don't see a lot of policies on the horizon globally that are specifically focused on microplastics, and I think that shows a gap where we need to do more science to policy research to talk about, okay, what are the sources? We were just hearing about some of that from Ezra, like what are the sources of microplastics and what are the policy interventions that we can put in place to address those? We do see more and more policies about preventing macroplastics from entering the environment. The big battle is really about, are we going to address plastics as a waste management issue, or are we going to address it upstream as a production issue? And in production, I'm talking not only total volumes, but what are the additives, what are the chemicals that are going in, what are the formulations, right? And then what are the uses, right? Plastic is this obviously extremely durable material, and we treat it as something throwaway. So we shouldn't be surprised that it's out in the environment. And California can have a very strong impact in all of this. So I would encourage you all to get involved.
Thank you, Dr. Tangri. Yeah, I think we can have like a quick, if anyone has a very quick question, otherwise we can, I'll remind everyone that we have panel discussion and you can also ask these questions there.
Hi, great talk. I'm not entirely sure how trade regulations work, so this might not be the greatest question, but I'm wondering, because you said that countries might use that as leverage to get other countries to fall in line with plastic regulation, could it work the opposite way? Could some plastics do countries force other countries to not regulate as hard?
Yeah, that's a really good point. I guess I can give you two answers. So first of all, I'm also not a trade lawyer. I have some familiarity with WTO and those processes, but generally, so trade is regulated both by the WTO as well as by bilateral and other multilateral agreements like NAFTA, right? Historically, that has not been the case. But these days, kind of all bets are off, right? So who knows? Maybe. Don't suggest it to anyone, please. I do think that it would be harder to put something like that in writing. I think it would be relatively straightforward to say, well, we're going to put a tariff or we're going to put a ban on plastic resin and plastic products from certain countries because they are not parties to this treaty. If you were to go after countries and say, well, you have to take yourself out of the treaty, or we're going to put tariff. I'm not sure exactly how that would work, but yeah, threats and promises all work in geopolitics.
I just say that because in Canada with the tariffs right now, like a lot of the companies that align with some of the regulations more would get more benefits if like they cooperate.
Right. So, yes, I don't think I've seen a case where tariffs have been used to push a deregulatory agenda, but yeah, nothing's off the table, I guess.
I think that's a good
All right,
point.
thank you.
And thank you, Dr. Tangri. I know we're a little behind, but we'll take a 10-minute break. I think everyone needs it. And then we'll start the last talk. Thank you.
Seats now. It's now the. Okay, can we please slowly make it back to our tables to finish up? I just wanted to make one comment before our last keynote speaker. We've had so many amazing speakers today talking about the full range of microplastic research, effects on ecosystems and organisms, monitoring, mitigation, risk assessment, international treaties. And so please stick around for a discussion with all of our amazing speakers at the end of this talk, where we can kind of bring all these amazing minds together and talk collectively about what we learned today. So I know it's been a really long day, and thank you so much for sticking around.
So it's now my pleasure to introduce our second and final keynote speaker, Dr. Chelsea Rockman. Dr. Rockman is widely recognized as a leader in microplastic research, known for both her scientific accomplishments and the real-world impact of her work. Her outreach efforts have significantly advanced public understanding and policy action of plastic pollution. She frequently advises policymakers at local, national, and international levels. Through these efforts, Dr. Rockman embodies how research and public engagement can come together to drive environmental change. She's an assistant professor of ecology at the University of Toronto and co-founder of the U of T Trash Team, a science outreach group which has received multiple awards for its environmental impact. Her keynote today is titled The Fate of Microplastic in a Whole Lake Ecosystem Experiment, Informing Monitoring and Risk Assessment. Please join me in welcoming Dr. Rockman.
Thank you for the introduction, and thank you for inviting me. It's nice to be back. I lived here 10 years ago before I moved to Canada, where I did my PhD, did a lot of work in California, continue to do work in California with SFEI, with SCORP, and I've actually been really lucky to translate some of what I learned here up there, and hopefully vice versa. So I'll talk today a bit about the work we're doing at the Experimental Lakes area in Ontario, but I will try to keep it all relevant to what we're doing here and globally as well. So my presentation today will talk about the history of the Experimental Lakes area so that you're a little less uncomfortable with the fact that we are polluting a lake. We'll introduce the Plastic Project. We'll talk about how it can inform FATE, how it can inform methods for monitoring, including how we've tested some of the monitoring sampling protocols that Ezra mentioned earlier. So we tested one in our lake, and we'll talk about how what we are learning can inform risk. So I'll start by introducing the ISD Experimental Lakes area. By show of hands, who's heard of it? Like half of you. Awesome. Okay. So this is in northwestern Ontario. For people who really love to fish, it's near Kenora. It's a 22-hour drive from Toronto, north and west. It used to be run by the government, so they set this land aside about 60 years ago. It has over 50 lakes. They are set aside to be used for research, both long-term monitoring, but also for manipulative experiments. The government almost shut it down when we had Harper, but they did try to shut it down, but ISD, an organization, took it over and kept it alive. It was started by the late, great Dave Schindler, who has done amazing work and really, I think, led to the success of the Experimental Lakes area. A lot of work has been done there just simply to understand how global change impacts lakes, but also over the years, these lakes have been used for manipulative research to use whole lakes to understand how contaminants impact whole ecosystems. This is hard to do in nature because there's lots of stressors, as Wayne told us, in, say, for example, if I'm working in Lake Ontario or San Francisco Bay, how do I know if the impacts are from the microplastics or from all the other stressors there? So that's one real benefit. You do have some control. Also, in a laboratory, everything's really standardized and controlled, and you sort of miss some of those ecological surprises. So when Wayne was telling Ashlyn, oh, I'm not surprised because you basically have a different lake 3 months later, there's a lot of environmental variability and realism, and it's interesting to see what do you see in nature. The ELA has been around for a while. This is a really famous picture from up above one of the lakes, Lake 302. All the lakes have number names, where they were trying to use them to understand how does eutrophication cause harmful algal blooms. They weren't sure if it was the nitrogen or the phosphorus. They split a lake in two. Basically, there's a curtain you can't see that goes all the way to the bottom. Carbon, nitrogen, and phosphorus on one side, the green side, and just carbon and nitrogen on the other. This study got phosphorus out of our detergents, and today we have less harmful algal blooms, but of course that problem's not gone. Similarly, acid rain was discovered in one of the situations in this area. I think it was probably also discovered in other places. A lot of work was done here to acidify a lake and understand it, and also that informed the acid rain treaty, and acid rain is barely a problem today. So yes, a lake takes one for the team, but it gives us a lot of information to inform change. So I believe in it. And when I moved to Canada, I was like, how do I work there? So today I am leading the Plastic Project with many, many other professors, and I'll show you who they are in a minute. But basically, we are adding microplastics to a lake to understand the physical, the chemical, and the biological fate of microplastics, to understand how microplastics impact ecosystems at all levels of biological organization, to also understand how they impact ecosystem processes. And when we're done adding microplastics to the lake, we can use it to understand recovery, both natural attenuation, degradation, but also testing it for active mitigation processes. So here is just a picture of all the PIs working on the project. I don't have pictures of all the many, many students, including Ashlyn, that have been part of this project. And then Des Langenfeld was a master's student on the project and now is our project coordinator. You probably recognize Suzanne Brander in the middle of the screen, who's here today and also part of this project. And there's probably some other faces you recognize as well. Our timeline is long. I call this my career project, and I'll retire when we're done. Not really, but right here we are now in 2025, but we started in 2018 dreaming of this project. In 2019, we started background sampling to understand contamination of microplastics in the lake, which is low and mostly fibers. We started baseline sampling our reference lake and our experimental lake in 2019, used our experimental lake for some controlled mesocosm, in-lake mesocosm studies to get some ideas of how we wanted to do our whole lake, and started adding to the lake in 2023. We add for 3 years during the ice off season, which we don't have to worry about here in California, but in Toronto, the lake only just melted on Monday. Not in Toronto, but in northwestern Ontario. So our field season starts now and it ends at the end of October. So we will add this as our 3rd year, and then we start the recovery phase. Ashlyn introduced the microplastics, but here you can see some of the additives that are associated. Well, actually, I didn't put the additives in there, but if you have questions about them later, I can tell you. They are meant to mimic actual products, and you can see the size distribution there is about 20 to 1400 micron in size. Because we toll ground them, the size distribution is a little artificial. The average size is around 100 to 200 micron. So I'm going to walk through results from 2 different experiments that we've done there. The whole lake I will talk about, but I'll also share some results from one of our pelagic corrals. So these are in situ mesocosms, or we call them limnocorrals. They are 10 meter in diameter, so they are large. Think about an above ground swimming pool kind of put into the lake. It has a bag down below, so it is cut off from the lake, so the microplastics in there stay in there. We used an ANOVA design with 3 replicates of each of 3 treatments, 1 a control, and 1 of plastics with additives, and 1 of the exact same polymers without the additives. So in this year, we were interested in how effects vary between plastics with and without the chemicals. I'm not going to talk about the effects from this experiment today, though, just the fate and how that informs monitoring and what we expect to see in the whole lake. The amount we added, the loading amount, is 29,000 particles per liter. That's not the concentration we see. Microplastics don't homogenize or homogeneously mix in a water body, as you know. So that's the loading concentration. I'll show you how it moved around there in a bit. For our whole lake experiment, we're using a BACI design, before-after control impact. On the top, we have Lake 378, our experimental lake, and on the bottom, we have Lake 373, which is a reference lake. We sample them both before everything relevant to organisms, the microbial community, the phytoplankton, the zooplankton, the fish community, et cetera, as well as water chemistry. And then similarly, we continue to do that after the additions. Ashlyn introduced this, so I don't have to, but we are adding the microplastics from a boat using Transex to try to add it as homogeneously across the surface as we can, so any distribution we see is not due to how we add, but instead due to physical processes, or biological, but mostly physical. The annual loading amount is 330 billion microplastics particles. We are simulating stormwater input, so we know the amount of runoff that enters our lake each year. It is a headwater lake. We use that to multiply it by a concentration reported in the literature that's quite high, 1,000 microplastics per liter. We add it bi-weekly, so every 2 weeks, so 12 times during each season, and it is equivalent to about a 77 kilogram. Well, it is 77 kilograms. That tends to be about the mass of an average man, I find. To put it into better perspective, it's about a cup going into an Olympic-sized swimming pool. So I'll start by sharing with you some results from our pelagic corral experiment, just to show you what is the fate of the microplastics within this system and what does it tell us in terms of how we think about the exposure landscape, meaning the exposure relevant to organisms for risk, and also fate, thinking about where we should monitor. And I think that a lot of us think about our beakers in the lab as we're going to add the plastic and it's going to mix around, and so the particle concentration is what we add, but it's really not what we see in a lab, and it's not what we see at all in a lake. So here is just the surface of the lake after adding the plastics. This experiment ran for 9 weeks. Here is the amount at 24 hours, 5 weeks, and 9 weeks. So you can see even after 9 weeks, there is a surface slick of microplastics at the surface. And so the next graphs I'm going to show you are both the total amount of particles over time on the surface, broken down by polymer type, and also the total, as well as the residence time. So the inset graph, first of all, has the total amount of particles on the y-axis and the time since addition on the x-axis. You can see it broken down by polymer. Most of what's there is the polyethylene, which is buoyant, so no surprise there. And you can see it decreases over time, but sort of hits what I don't know that I would call it a steady state, but it seems to slow down. The graph on the back part that's kind of busy is the residence time. So the black bars is the amount of time it takes for the 95% of what we add to leave the surface. And the gray is the amount of time it takes for 50% of what we add to leave the surface. The reason the bars are so big in certain cases is we sampled at 5 week and 9 weeks. So that black bar says that sometime between 5 week and 9 week, 95% left the surface. So you can see that for the polyethylene, the residence time is really long, but for the other 3 plastic types, it is much shorter, up to a week for some. For the PET, that's really dense, only a few days. And not a huge difference in this case relevant to size, maybe because it's sticky and they stick together, I don't know. But in the water column, that's quite different. The residence time in the water column is short. Most of the plastic has left the water column within a couple days. When you look at the inset graph, now you're looking at concentration on the y-axis. You actually see this spike at 1 week, and I think that's when a lot of the particles leave the surface and start raining down. Then we detect them in the water column. And here you can see the longer residence time, longer than a day, is only for the smallest size fraction of the polyethylene. And actually here I'm only showing you to 53 micron. I'm not showing you the smallest particle size fraction. We're still in the process of measuring it. Now, we only sampled what was in the biofilm on the mesocosm walls, which could be kind of similar to like rocks on a shoreline, or when you showed the macrophytes in the tide pool, or I don't know if you call them macrophytes in a tide pool, but the algae, similar to that. And then we also looked at the bottom. So we put basically sediment traps on the bottom of the corrals and then dove down and collected them at the end. So you can see at the biofilm on the walls over the 9-week period, we do have microplastics. It's mostly polyethylene. This is in particles per meter squared on the bottom. So in this case, we had these wall strips along the wall, and then we sampled them at the end. And on the bottom, you can see quite a bit of microplastics, and most of it is the PET, less of it is the buoyant polyethylene, and I can tell you from watching it that the PET went down to the bottom immediately upon addition, so it really wasn't a big part of the experiment, the PET, although some fish do feed from the bottom. So if you try to look at a mass balance here over time, here I'm not showing you the biofilm and the bottom, I'm just showing you them together as the black in the donut at the top. You can see how the surface amount changes over time, and you can see how the amount in the water column changes over time. You can see that of everything we added, only 2.5% was ever in the water column, and that was only at that 1-week period. Most of the time it was less than 1% of what we added, so the zooplankton had the lowest exposure. On the surface, you can see that in the beginning there's quite a high concentration, which could be important for organisms that feed at that surface area, and then that eventually goes down, and the bottom is obviously quite important. And the biofilm on the mesocosm's walls also had more than 1% of the plastic, so important for organisms that feed there. So overall here, I'd say what we learned and what that would help us predict when we get to the whole lake is that the residence time on the surface is longer than in the water column, varying by size and density. So we would expect to have perhaps a surface slick on the surface of the lake, but remember, no, it's no longer in a limno corral, so it might take off to the shoreline, as Ashlyn showed you in her talk. Most of the plastics sink to the bottom and actually fairly rapidly, so the bottom is going to be important for both the organisms, but also if we see burial for eventual natural attenuation, perhaps. And plastic is likely to accumulate on the shoreline based on what we saw, I think, in the biofilm around the sides, and so that made us start to think about the biofilm on the rocks and the woody debris and the macrophytes, and Ashlyn mentioned that now we have a student looking at that, so Ren is looking at that, and he is looking at more matrices than Ashlyn did and trying to really understand if he can figure out what's in these different, like on the rocks and the macrophytes, and scale it up to the whole lake. So in terms of that, that means we are eventually trying to do a mass balance. So I will start to show you some preliminary work in terms of what is the fate of the microplastics. I'm only going to show you within the first 13 days, just a sneak peek. The nice thing about the first 13 days is it's the time since the first edition and before the second edition. Once we add again, we don't know if what we added, let's say in 3 weeks, was from the second edition or the first edition. So it's more controlled. So I've been working on this mass balance right now with Matt Hoffman, who's a professor at RIT, and our PhD student, Francis Nwama, who's at University of Toronto. I'm just going to show you data from the water column, from sediment traps. So we're using that as basically a proxy for how much is on the bottom and what has washed up on the shoreline. So we were looking at beaching from the beginning, but we weren't looking at those matrices Ashlyn showed in the first year. So you can see the different parts around the lake at which we sampled, 5 stations for the shoreline, 6 for the pelagic, and 3 for the sediment traps. The data I'm going to show you is scaling up. So basically it's going to be the average in those samples, and then scaled up across the entire lake to look at how many of our plastics that we added were in each compartment. I'll start by showing you the totals, just the trends, and then I'll show you the mass balance by percentages. So for the water column, I am showing you this split by thermal layers. So the epilimnion on the top, the meta in the middle, and the hypolimnion on the bottom. And that's the shading. You can see the total in the top, your top left. And then you have the polyethylene, the polystyrene, and the PET. You can see that most of the microplastic, again, does leave the water column after 1 day. And the majority of what is there is the polystyrene, which makes sense because that's neutrally buoyant, whereas the PET and the polyethylene aren't so much in the water column, even though they're there a bit. Next, I'll show you the sediment trap data, and here you see a video of us roaming around with our drone. On the bottom, you can see those blue particles falling down and eventually sticking to the rocks. But here you can see that the PET by 1 day is pretty much as the PET is down there, and the polyethylene and the polystyrene climb over time with more of the polystyrene on the bottom than polyethylene, and that is because the polyethylene is primarily on the shoreline. After 1 day of additions, we don't see as much on the beach. This picture is not what it looks like all the time, I promise you. If you go to the lake after even 2 days of addition, it looks like a normal lake. You don't see the microplastics. If you look close on the shoreline, you might see yellow rings around the rocks, but the pollen looks almost identical. This is what happens hours after an addition, but this is, I think, why we don't necessarily see all that plastic beached on day 1. We start to see it later when you get winds pushing the water or the plastics up onto the beach, and you see that increase throughout the 13 days. So then if I show you what this looks like in terms of the mass balance, here you can see at 1 day, the percent of all the plastic we added that is in the water column, only 12%. Most of it is polystyrene, neutrally buoyant. At the bottom, almost half. Most of it is PET, but some polystyrene. And at the beach, only 2%, and most of it is polyethylene. And by 7 and 13 days, you can see those numbers increase in both the bottom and the shoreline, but the water column goes down, and it's pretty much the same at 7 and 13 days. And if you do the math, we're accounting for about 78%-ish. I didn't actually count it, so if you're like, it's 79, I don't know. But it's somewhere between 75 and 80. And so then the question is, where is the missing plastic? So here you're looking at graphs that are actually the same data as what I just showed you, except for that now you have the day since addition on the x-axis. Each polymer is you can see a different graph. The total microplastics is on the y-axis, and that dotted line is the total amount. So the PET hits the total amount in the sediment, the brown matrix, so we're accounting for all of it. But if you look at the polystyrene, you can see a big gap, and there it's possible some of it is going out with the outflow. Our outflow is a trickle, and yes, it does lead to a lake downstream, but it's about 1 kilometer away, and it's a trickle of wetland. So I'm not sure it went out and then went into the other lake below it, 379, and we're actually monitoring that lake and haven't seen microplastics. It is likely in the wetlands in that area, but it very well may have left. And then the polyethylene, I think what we're missing is probably a lot of that, and some of the polystyrene as well, is what Ashland and Wren have been digging into on the shoreline. So what does this mean? Basically, so Inga, I have to tell you that one of the things you taught me when I was in grad school is you used the word pseudo-persistent to talk about, I can't remember what contaminant, and it stuck with me. And this is actually the way I think about microplastics in the water column after these experiments, is that I think microplastics are actually pseudo-persistent in the water column. The only reason I think we keep finding them there at increasing or static concentrations is because we keep adding it. And I think if we turned off the tap, we would actually see those concentrations in the water column go away, and where we should then focus in terms of remediation or just thinking about risk would be washing up on the shorelines, which is why I got excited about the tide pool data, but also I think sinking to the bottom and thinking about the benthic organisms. So I think a lot of those buoyant plastics eventually wash up on beaches. Some people think that a lot of the plastic mass balance is on beaches when you talk to some of the people trying to solve the macroplastic mass balances at least. They also likely accumulate in biofilm on rocks, woody debris, and macrophytes. Those are often ignored and really important in terms of the base of the food web. And this should inform, I think, what we understand about fate and also exposure and risk. A lot of the risk data, Scott can tell you in Tomex, is for these pelagic species. That's important because the tap is very much still on, but we definitely need to focus on those benthic organisms or those littoral shoreline organisms because they're probably getting the most exposure. Okay, so that's fate in the lake. The next thing I want to show you is actually testing some of these monitoring methods we've been developing for the state of California, using our whole lake experiment to do it. It's actually fun to go last, not because you're all tired and probably want to go home because it's Friday, but also because I get to piggyback on what everyone already talked about. And so I think Scott mentioned that pumping is maybe ideal because it can reduce the contamination. You can also get smaller particles and you have less particles seeping out the manta. So the method that is suggested in the California monitoring work group, which is led by way more people than Eden, Claire, and Brenda, but we were the ones who tested it at ELA, so that's why they're here, suggests a pump going through a sieve stack and taking as much volume as you need depending on the concentration, so enough volume to get a representative sample. We tested it next to a manta, which we suggest as a benefit if you want to take both as complementary. Thinking about what we saw on the shoreline in the limno corral, I thought, oh, I'm going to see a totally different answer for the manta trawl because maybe the surface is a different matrix. So I was really interested in what we were going to find, and I can tell you here this manta trawl has a 53 micron mesh, and so it's directly comparable to the bottom sieve, which is a 53 micron sieve. So we took it out to the lake one day after addition when we knew the concentration was around a particle per liter, and we tested 3 different types of pumps that cost different amounts of money and pump at different rates: a gear pump, a peristaltic pump, and a submersible pump, as well as a manta. We took 20 liters and took 3 reps. Then we asked, does tube size matter? Which seems really mundane, but it might. If you're looking for particles, is it possible that the physics of the amount of water coming into the pump makes some particles kick out? So we looked at 2 different size tubes, and then we did a volume ramp up to see how much sample do we need to get a representative sample. We could figure this out mathematically, and some people have. There's a great paper called the RSVP tool, but we wanted to see it in action and actually see if we could plot it and get what we expect. So we sampled 10, 20, 40, 80, and 160 liters. So first we saw, and when I show you these plots, on the left is going to be the total amount of microplastic, and then on the right will be the different size fractions. We didn't see any significant difference between equipment, which was really nice. That includes the manta. So in this case, I think because of the wind mixing and or the amount of washing up on the shoreline, the surface in this case was very similar to, I should tell you, a depth integrated sample from the top, just under the top to 1 meter down. We also saw no difference in tube size with the peristaltic pump. Yes, it looks like maybe you have more particles with the larger tube, but it's not statistically different, so tube size is okay, as long as we suggest you have 6. We decided it should be at least, I think it was 2 or 3 times the size of the largest plastic you're trying to encounter. So for the volume ramp up, actually we saw what we wanted, which was nice. Now remember, we are only looking for fragments, so it could be that fibers complicate things, but in this case here, on the top is in concentration and on the bottom is the total amount of plastics. So once you hit the concentration that is representative or more accurate of what is in the lake, you should have a flat line at that concentration across the volume, but when you have the count of particles total, you should see a linear trend. And so 10 was too low, which we expected, and 20 was good and up. And so now really what you're saying is if the sampling methods are all the same, then you can choose based on how much time you want to spend sampling and how much money you have. So here's just a comparison of how much time it took for sampling 1 liter. The Manta's obviously very fast. I should say that the submersible pump is my favorite. It's the fastest, but we ran it at different depths, so that's why you see such a range. And then you can see the price there in USD. So a gear pump is really great if you're on a low budget. It also goes really deep if you're also on a low budget, because the peristaltic does not, but it's also the most expensive. Okay, so in my final minutes, which hopefully I have them, I just am going to give you a sneak peek at some of the ecological effects in the lake. And actually, I know Scott talked about this, and I feel like at least one other person talked about this, that for microplastics, we think food dilution is one of the mechanisms of effect that is related to a physical effect of the microplastics. And I do think we're actually seeing this in our study. So in our pelagic corrals, actually I lied, I'll show you some effects from there. These are yellow perch. This is the size of the fish 9 weeks after exposure. And here you can see we do have a significantly smaller mass of fish exposed to both types of microplastics. And so this suggests that maybe there is a decrease in growth, potentially from a food dilution effect, or maybe even changes in the nutritional value of the lower levels of the food web. So Garth Covington has been digging into this. He's looking at the fatty acid ratios and amounts within, in this case, the biofilm on the walls, the plankton and the algae in the pelagic, and the detritus on the bottom. These are NMDS plots, so the closer the dots are to each other, the more similar they are. So you can see that the blue and the light blue are close to each other. Those are the 2 plastic treatments, and the control has separated, suggesting that there is something going on there in terms of the plastic changing the fatty acid content and or amount within those lower trophic levels. So that's interesting, and that suggests that the food web has potentially changed. And I can tell you that for some species of zooplankton, even though I'm not showing it, we did see population level effects where they had a decrease in reproduction. So this makes you wonder, what do you see in a whole lake ecosystem? I'm not showing you much data because we're really trying to wait till we have multiple years of data, but I will show you quickly just some preliminary results for the phyto and zooplankton. This is led by Maddie Milne, who is a master's student with Mike Patterson at the University of Manitoba. And here you're looking at the biomass of the phytoplankton over the years. 2023 is the year we started additions. I'm not showing you Lake 373, the reference lake, but I can tell you that we don't have the same pattern. Here you can see for 2023 a reduction in the phytoplankton, suggesting that something may be going on in terms of the amount of phytoplankton in the system, which you would expect could then impact the next level, which would be the zooplankton. Although the phytoplankton recover in 2024, in 2024 we do have this decrease in the biomass or the total abundance of zooplankton per liter, again in our experimental lake, and although I'm not showing you, not in our reference lake. So this suggests it's possible that the zooplankton had now had that effect from the phytoplankton before, but have released the phytoplankton from predation, and now the phytoplankton could have come back. Or this could be total speculation, and it could be a 1-year trend, and in the future we won't see anything at all. So we will see. We did not see an impact to the population of Chaobarus, which are the next level up. It's kind of like a zooplankton, but they can swim, so it's arguably whether they're plankton, but they eat the zooplankton. No effect to their population, but we do see a decrease in the size of the Chaobarus post-microplastic addition in the experimental lake, and the mysis we used to compare in Lake 373 because we don't have K. arboris in 373, and we don't see a significant decrease in the size there. So this suggests maybe we are seeing food web effects. I don't know if you remember the synthetic estrogen study where Karen Kidd saw a crash in a fathead minnow population, which did ripple through the food web. For us, this would be for a different reason. If we are seeing what we're seeing, again, early days, and we will to be continued. But I would say overall, we may be observing signs of indirect effects we likely could not detect in laboratory studies. Microplastics might affect the quality of phytoplankton and zooplankton, which may affect growth, condition, and reproduction. And I think these longer-term studies at larger scales, while they can't really inform a risk assessment, they can certainly sort of ground truth it or let us see, okay, is what we might expect from these simpler, they're not simple, but from these other models, what we do see in a whole ecosystem. So thank you. I have no idea how I did on time because it was running the whole time. Do I have time for questions?
Thank you, Chelsea. Great to kind of meet you and see you. I've read a lot of your work.
Thank you.
This whole conference has been awesome. I feel like I'm meeting so many of my idols. Can you remind me what the 3 plastic types were in the experiment? It's PET and then...
It's PET, which is our dense one, polystyrene, which is neutrally buoyant. It's not the foam, it's polystyrene, high-impact polystyrene, and then polyethylene, which is our buoyant, all
Gotcha.
fragments.
So can you speak to why some of the polystyrene and polyethylene are sinking?
I think it's because over time they grow biofilm and they become more dense. So when we first add the plastics to the environment, they behave like brand new pristine plastic, and the polyethylene sticks together and floats to the top and wants to go everywhere. But eventually, as they grow biofilm and they weather, which is why I was asking Suzanne about what she saw earlier, they do change behavior, and we do see them even in the sediment traps, the polyethylene, but it takes a few weeks.
Gotcha. And so you are confirmed seeing this biofilm on those particles that have sunk that were initially floating. Like it's a fact.
It's a fact, but we haven't measured it, so I can't tell you like this much biofilm makes it sink, but there are students who are looking at that. We are also seeing burial quite deep in the sediment, more than you'd expect,
Awesome.
which is not what you asked, but also surprising.
Cool. Thank you.
Wayne.
So I want you to, I would love to do the risk assessment and then you do the experiment. See how good we are.
You want to, wait, you want to do what? You want to do a risk assessment?
And then you do the experiment.
Please do.
Because the part is that if we have everything that's really well understood, we should make some reasonable predictions, but there'll be changes for size and stuff. And also, it may also point out the kinds of things that we're finding could be important, and maybe you didn't or couldn't, or what couldn't measure them
Right.
a priori.
Yeah.
One of the reasons I do what I do is that back in the 80s and 90s, I ran lots of Taubes microcosm experiments. So all those dynamics and stuff, you know how they get in your head?
Yeah.
And you think about all these things interacting. I think this is a great case. Much as Karen Kidd's paper demonstrated the persistence of effects due to the estradiol. Of course, you lost some of the older age classes because of the way you changed the dynamics. And even though the estrogen was very likely gone, you could still see those gaps for a long time in those experiments. That's why I put it in the textbook.
Yeah.
Is that it's such a classic persistence of effects long after the contaminant's done.
Yeah.
And everyone's making the assumption that, well, the chemical's gone, it'll be better.
Right, which we
No.
get to start to follow that next year.
Yeah,
which
we'll
we're
see,
excited
we'll
about.
see.
But if you want data and you want to do a risk assessment and then compare it to what we find,
Yeah.
we would love that.
Yeah, that'd be cool. Yeah,
Okay, one more question and then we will move on the discussion.
I'll keep it quick. Thank you. Great talk. Thank you very much. You mentioned something about burying of the microparticulates in the sediment. What do you think is driving that?
It's a good question. I don't know. There are papers that are showing microplastics way below the depths you would imagine if you have a sediment core where you know the years of the sediment core. So this is Hannah's data showing the microplastics down to 10 centimeters, and this is just after the first year, I think in August, after a certain amount of additions. I don't know what's driving it, and I think it's something we want to understand, so we're going to try to do some experiments on that. I don't think it's bioturbation all the way that deep, but I don't know. Yeah. Mm-hmm. Okay. Mm-hmm.
Can I please have all the speakers, including the student speakers, come on up here for our panel discussion? We might need a couple more chairs.
They're all on? Yeah.
Microphones so that we don't have to pass a mic all the way down 10 chairs.
Oh, that's cool. Yeah, yeah. Yeah, we want to have like, how many microphones full? If we have two in the audience, maybe, and then 5 total? Shall we do? We'll have. We'll just put them at the end and the middle.
Testing 1, 2.
Hello.
Hello, hello.
I think while we're getting the last microphone set up, we can start. So I think we're interested in the key questions, the open questions that we still have from this microplastic symposium. And I would just want to start with the seeding question maybe. What do you think or what knowledge gaps do you think in your field maybe should be prioritized for funding in the next 3 to 5 years maybe?
Well, it kind of depends on what's happening in terms of, agencies existing and that sort of thing, but we won't go into that. But in an ideal world, it would be wonderful to better understand translocation, to have funding for more long-term experiments. It would be great to have more multi-generational experiments. There have been a few done in invertebrates. I know that Chelsea's lab recently published one on fish, but looking past just the F1 generation, thinking about how long do these effects carry out for different particle types, for mixtures, better understanding what's happening with fibers, and I guess all particles in terms of the chemical and physical interactions. I feel like we can understand one or the other separately, but we don't fully understand how they work together. So that's my wish list.
I would also just add more research on the nanoplastic side, both in terms of the toxicity, but mostly in terms of environmental monitoring. We have very, very few nanoplastic monitoring studies. And when it comes to risk assessment, we need a model that is able to account for the non-particle based toxicities in the same framework. So understanding fugacity and how chemicals interact with biota from the particles back and forth, and then also the pathogen in a synergistic way. We don't just see additivity effects with these different stressors. They combine.
In terms of management, I think a lot of more closer to source control things need more research. So remediation has got actually quite a bit going on, but thinking about a little bit upstream. So for tire particles, things like how effective tire particle catchers on vehicles can be, that sort of thing. For fibers, thinking about how to change how much they're shed from various textiles and which textiles are the biggest shedders, that sort of thing.
If I can piggyback on that, I do think from a policy perspective, we need a lot more work, as we touched on this, on what are the strategies to reduce microplastic emissions, and over the whole range. And I think I would encourage us to think even more upstream, right? So if we've got tire wear, okay, reformulating tires and things like that, but also are we talking about moving to different forms of transportation so that we're not moving a 2 tons of metal around with us to get from point A to point B? Paint, right? Paint is on everything. What are the alternatives to paint? And again, some of it may be reformulation, some of it may be alternative materials, but I think we need to think a lot more in terms of systems change, and what are the policies that can drive systems change.
I just want to add one thing to that, which is that this is for not just microplastics, but it would be multi-benefit. So paints contain lots of other problematic compounds like PFAS. Tires contain lots of other toxic contaminants, and also produce greenhouse gases. And so this isn't just a microplastics question, but microplastics is one piece of a larger whole.
Sing it.
I think one thing that many of us are acutely familiar with is how labor-intensive and expensive microplastics research can be. And so I think one thing that I would like to see moving forward is research on development of new technologies and new ways to make microplastics research less expensive and more efficient, especially considering just how complicated it is compared to some traditionally studied contaminants that might help us get some of these studies out faster so we can actually tackle this problem efficiently.
To piggyback on that comment, I really think that it would be interesting to start harnessing some of the power of AI to really help with some of the time efficiency, potentially with some of the kind of identification and the more mundane, time-intensive tasks related to some of our work. So potentially looking to the future that way.
I think also with just lab work generally, I feel like we all use a lot of plastic as is. So I think it would be interesting to also start working on techniques where I feel like I always tell people when I do plastic like mitigation stuff or I recycle and stuff, I'm like, as a plastic pollution scientist, if I don't do it, I don't know who's gonna do it. So I figure those kinds of solutions as well.
I'm also going to piggyback on what Zeke said. Specifically for tire wear particles, we can't use Raman spectroscopy or FTIR spectroscopy to get quality spectra of individual tire wear particles just based on their chemical composition. So there's really a need in that space to be able to find some sort of technique that can help us identify them chemically.
I showed you, we don't have the data sets. So is it hard to rank what's the chemicals that are causing effects? Because we don't have the numbers. So this is, Isra, I think $1 million sounds like a lot. But how much have we, all the PIs here, how much have we spent in our careers? $1 million isn't that much. Especially consider what it costs to go to a contaminated site, a Superfund site or something similar. You realize how much money they spend on characterizing a site? And I've never seen an effort comparable to that for an area that we have some reasonably good idea that is contaminated with microparticles. I don't mean just plastics, but microparticles in general. I have not seen that characterization, and maybe someone else here has, but until you have the data on exposure, I don't know what to tell you guys to use for doing your toxicity experiments. It's like we're blind here. And part of this is like, we do all this work, like we had the seed and surf data sets over years and years and other data sets for the Bay, and also San Francisco, and for the Delta and for the Bay. Well, wouldn't that be great to have 20, 30 years worth of data, this kind of stuff, sampled the correct way with modern data analysis tools in mind, and also would force everyone to get better and more efficient at counting the particles. Until you put that effort into it, my uncertainties just blow up in my face for the plastics compared to bifenthrin and everything else that we know so well. And you'll find that the more you do the sampling, the better you'll get at it. And I don't trust AI. I mean, my daughter's an artist, and my daughter's an actual artist, and you look at the AI stuff and going, oh God, this is so bad. But but I also use it every time I do a tree search, every time I do a literature search, I ask a question and I get faster answers because I ask exactly the right questions. So I'm not saying don't use it, it's just we gotta get better at it. Give me the numbers and it's easier to do the risk.
I just want to clarify something for those of you who are maybe not familiar with all the acronyms. CDEN is the California Environmental Data Exchange Network, and it is a really great resource of lots of environmental monitoring data. However, it is not particularly well designed for microplastics, and this is maybe a larger, yeah. So this is another need, is a way for us to gather all the microplastics data in one place. This is something that's actually being worked on right now. But just like how Tomex, it takes a lot of effort from lots of people to gather the data, publish in papers, and put it all in one place. Same thing with monitoring data, and it would be great to have better systems for those things moving forward.
I think CDEN is very good, designed for a while ago. We can do a much better job. And in some of our reports, we've talked about the issues with it, is that here's where the benthic sample was done, a kilometer away is the water temperature and everything else. Those two things are not necessarily related to each other. I could see that for all kinds of things. It's a kilometer is a long way in the Delta system. In a lake, it's a big, big way. And we can do so much better job of measuring these things now. And to also think long-term, but no, I completely, and I'm in California, so I assume people know what they are. And I was incorrect. Uncorrect is what all my kids would say. Dad, you're uncorrect.
Do we have any questions from the audience or from Zoom?
Hi, so we are the toxicology lab. So part of the reason why we started with the long-term monitoring in the reserve was to set up the baseline of what kind of particles we're seeing, because the idea is to later do the toxicology testing with the particles that we see most abundant. But from what I'm hearing here is that we also need that reference microplastics that we are gonna compare our results to. So that we have a library, or if you guys know of any reference microplastics, or what are the efforts on those reference microplastics that we can use for toxicity testing.
There's a couple resources out there. There's a company, I believe they're based out of Germany, called Microplastic Solution, and they sell environmentally realistic very well characterized of microplastics reference standards that can be used for quality control, for doing analytical methods, but also for toxicity tests. And then the Coleman's lab out of the Netherlands recently developed basically a cookbook protocol for how you would prepare these environmentally realistic mixtures of microplastics from off-the-shelf components that you can buy. But it requires fairly complicated cryomilling techniques that Suzanne Brander's lab actually does, and I'm sure Chelsea's lab probably, some others. So collaborating with other research groups that are already making these mixtures might be your best bet, or buying it off the shelf from Microplastic Solutions or a similar source.
Has a follow-up?
Yeah, basically it's a follow-up on that. So if we're calling that like reference, that means that everyone in the entire world is gonna use the same mixture? Because ideally that's what I think will be the thing that will make things more comparable or more useful, or do you think that that's not necessary for this risk assessment that you are expecting? So if I use fibers from one lab and I'm comparing with somebody who's in Germany and is using polystyrene beads, how comparable will be our results even if we're using the same organism for the risk assessment perspective?
Well, the issue with microplastics is there's like a million possible combinations, trillions, quadrillions of possible combinations of different chemicals, shapes, sizes, particles. And so there's never going to be one reference material that is going to be universally applicable to every such situation in the world. So if you're trying to characterize risk for your specific environment and you want to do it in the most realistic way, you would recreate or obtain particles from that specific environment. Otherwise, you're going to have to be doing alignments.
Yeah, this is not a laboratory, this is not rat toxicology. Because we don't, actually the fish are cool, but if you're trying to manage the delta stuff, you're managing a dynamic ecosystem. So if you really want to do this well, you have to understand the trophic levels and the context and everything really well. We see this even in rivers are very close to each other in Puget Sound. Well, these should be pretty similar. Well, they are, except they're not. Some have different salmon. I didn't want to turn it on. Oh, that's right. Everyone down. Now she turned it on, so you're the problem. So it's really important to understand the context. The Skagit River is not the Nooksack River, it's not the Yakima, and so forth and so on. And we see this here in the middle of California too. They're all subtly different, and they're really important that they're different if you want to manage that. So you need to understand what you're extrapolating to. It's good to have standards, and I love standards as a standard, toxicant, a standard chemical to make sure that your organisms are performing adequately or healthy and stuff like that. But it's very context specific. We see differences in different branches of rivers and stuff, just depending. And in California, you have the same issue. We have really dramatic landscapes, and a few kilometers can make a huge difference, which everyone says now it's too hard. I teach 20-year-olds to do this stuff. But yeah, it's completely doable.
Just jumping in there, as you saw with my Tahoe project, we definitely had a little bit of a different characteristic in that regional area. And so I hope that one day we'll be able to understand maybe some of the risk factors associated with the types of mixtures that we're finding in an environment. So perhaps, even though I don't understand exactly what the physical characteristics of the particles that I collected and looked at are now, my hope is that someday we might be able to say, hey, look, we've got this combination of different maybe factors in this particular environment. We know that those are linked to certain health effects. And then, maybe strategies can be made specifically for that. But I agree. I think that the differences are very regional, and they'll change over time as well. So as consumer, preferences change and even industrial or housing changes in certain regions, it's going to change the type of microplastics we probably see in different regions. So while I would love to standardize everything to be completely the same across, the entire world to make sure that everything's comparable, everything is a little different, and it's a little. I feel like it's simplifying it to say that we could do that and make it helpful for us in understanding some of these risks.
Great. I know we have more questions from the audience, but just because it's on this topic of standardization, which is so important, especially when we're comparing different studies, what would the panel think is the biggest kind of bottleneck or limitation in standardization methods? Is it sampling? Is it extraction methods? Is it different mesh sizes? Contamination, different protocols to prevent contaminations? What can we do as a community to standardize and optimize our protocols so our studies are a bit more comparable?
I think Scott had a great point when you realized you had to get rid of some of those data points because the experiments weren't done correctly. That, I'm looking at, it's like, I'm a terrible reviewer because the first thing I do is look at their methods, and often in like 5 minutes I'm done. But do we actually have clearly standardized methods on how to do these toxicity tests, like ASTM? Environment and Climate Change Canada has actually a great standards group, and for their design of their experiments, their statistical approaches and so forth, they're some of the best I've ever come across, and very detailed. I love getting a new report from those guys. Oh, Wayne, here's another one. I haven't, and also as I was talking about, when I was doing QSAR work, quantitative structure activity work, we would go do this huge literature review, like on mutagenicity. We'd throw away 80% of the papers because they were not suitable, they were not done strictly enough and with enough samples and so forth, so we actually could do a statistical analysis of the results. I am so frustrated. And that's one, I think, one of the big advantages we had, Susan, between us and Stacy. We all did the same thing, the same kind of protocol. And I think there's actually much less data on microplastics, and I just heard that today, than what most people assume. They look at total number of papers. I always go 90%, let's take them away, and that's maybe what we have. It's, I don't know, your experience may be somewhat different, but I read those same papers and it's like so frustrating.
Yeah, I think for the sampling aspect, we have some standard operating protocols available now, and actually just a few weeks ago, Southern California Coastal Water Research Project published the SOPs for the State Water Board for sampling microplastics in sediment and biota. And we also just provided an SOP for how to sample in drinking water. We also have an online training and lots of resources that go with the sampling techniques. And those are based on protocols that they're already implementing across all of Canada, monitoring in many drinking water treatment plants.
Yeah, I totally agree with what Wayne and Scott have said about toxicity testing, about sampling, and I think we're beginning to get somewhere in those two aspects. If we're thinking about a bottleneck, I would say the extraction and analysis process right now is a huge bottleneck. I mentioned we're part of this interlaboratory study with the Southern California Coastal Water Research Project, and we are working with them on their sediment digestion and extraction method. And one sample can take 2 weeks to get through if you have challenges. There's density separation, there's sieving, there's the Fenton reagent, and the amount of time it takes, and also the limits because of availability of instrumentation. So we're realizing that there's so much variability depending on which instrument you use, that a lot of us are just flying down to Southern California and using their FTIR because some of us have older machines or some of us get slightly different results. And so I think, the only way we're going to fix that problem is with a lot more funding, so people are able to, we have more instruments available, or we have more centers with advanced instrumentation available, because that's really the bottleneck right now, is that if you have an instrument that's 4 years old, it's almost like having an old computer, right? It doesn't quite chug along as quickly. And so that's kind of where we are right now. And I think that's an area that we need a lot more focus on if we want to make strides in making our monitoring data and our extraction data comparable across labs.
I would also, I kind of come to this group with a perspective in, veterinary medicine and medicine in general. And for all of our testing and most of our diagnostics, we take an evidence-based approach in developing those methodologies to find best practices. And what I would say is, unfortunately, in this field, that kind of research is not very sexy and appealing to fund. And so it makes it a little bit more difficult for us to actually determine what the best practices are, so that we are choosing the right ones to kind of move forward with and kind of have that collaborative and comparative approach. So just throwing that out there for funding ideas, for sure.
Can I follow up with another veterinary perspective in terms of standardized methods? So a challenge that we face is that we're trying to detect microplastics and pathogens in the same sample. And the methods that have been optimized for microplastics are not always suitable for pathogens. So just as an example, potassium hydroxide destroyed the parasites that we're trying to count, and so we had to validate a different approach using pepsin digestion. And I hope it's okay when we go to publish this type of work, but I'm not sure what the microplastic experts feel about the fact that we do have to adapt and find other methods when we have different questions that we're asking.
I'm good with it.
Thank you.
We always have these issues, if you have analytical chemists, you can't take, you may have to just take subsamples and assume they're similar enough, because you have to treat them differently. It's just, this, almost wish we had a microplastic surplus site. So we actually, someone would invest the resources in this. It just reminds me of TCDD and PCBs back in the 90s. It was so hard to measure them, and now it's almost trivial. PFAS, there's a lot of different ones. I remember you'd have to go like a whole fish. Well, I don't know how much in different tissues, because it takes the whole fish.
I'm going to jump, I'm sorry.
Kind of trivial.
Oh, sorry.
No, it's okay.
I'm just going to jump in with a quick question. And the speaker before you, can you remind me your name?
Me?
Yes.
Karen.
Karen. Okay, Karen, thank you for your comments. And it was reminding me of something that keeps me up at night, which is this idea that tire wear particles are this underreported microplastic, but the methods for characterizing them are so different than micro-FTIR or Raman that we can use for other particles. So I'm wondering if anyone on the panel has any expertise using the same workflow to prepare samples that are adequate for characterizing or at least counting tire wear particles and traditional microplastics together. Like that, my hope and my dream is to be able to not leave tire wear particles out of future microplastic quantification studies.
I think if you want to do the same workflow, so not necessarily for automation, which I know is your dream, it's mine too, but if you're picking particles the way we've seen today, then those tire wear looking particles would go to Pyro GCMS, where you can similarly, like if you take particles to Raman or FTIR and you ping them with a laser and you get a spectrum, you can use it as particle by particle, but in a Pyro GCMS and get the signature for tire wear particles. So it's not so satisfying, but it can be done. But it does take longer. but I think even with the automation for microfibers and like if you're automating with Raman or FTIR, you see it reported in the literature, but people will tell you that their false positive rate is so high because like a microfiber is kind of like that, and every time it goes up, it counts it as a different particle. So with the automation, it's kind of messing up right now for tire wear and microfibers and potentially paint, which are all really important. So I think this comes back to we do need to put our heads together for better methods. Sorry, I'm like jet lag, getting tired. And I think for some of these like plastics that are probably really important because of the chemicals, the methods might be a little different, which I know makes you sad.
Just jumping in here because I do a lot of Raman, which is not great for doing tire wear particles. What I will say is that if we see particles that don't have a good spectra for other things, and we're seeing them very frequently, it does, I mean, you can use that information too. You can say there is a subset of particles here that I haven't identified that look very similar that we need to figure out what they are, and then you can follow up with additional testing. So just keep in mind that, you know, obviously if you kind of think through some of the problems, you can, you know, use the instrumentation that you do have access to get you to the right place sometimes, even though you might need to add some extra steps.
Yeah, and just to add to that, we do a similar thing with our FTIR. We have an ATR, micro ATIR, and if we consistently get this one match, it only gets to about a 50% match, but it's very reliably most likely a tire particle. And then we save those for later, and we'll send them up to our friend that has a pyrolysis GCMS. So you can kind of, you know, kind of roughly figure out what it is and then do better. Sometimes, yeah. Mm-hmm.
It's carbon black for Raman. I think we probably have time for one, maybe two more questions.
Yes, okay. So I have a question from Miami, from a student and also from an environmental activist educator. So the student wants to know, is this idea about microbes able to eat plastic all hype? Is it real? The other question is, how can communities collaborate with scientists to create citizen science projects to collect and analyze microplastics in the environment?
Because I actually have patents on the biodegradation of certain materials. And of course, they're of military interest, and so I worked for the Army at the time. And I also looked a lot with other kinds of biodegradation issues. Unless organisms can get ATP out of it, they won't degrade it down to CO2 and water. So it has to be a pathway where it's energetically feasible for them to do that, either through putting in an amendment or have something else done to it. That's what makes something biodegradable. And it's very difficult to do that. For years, they kept saying we could get rid of oil in the ocean because I have a bacteria I gene modified to eat oil. Well, the first thing it does is eat glucose or sugars or whatever else is there, so it switches. So it's very difficult to do that unless you have a very specific culture in a bioreactor under somewhat closed conditions. The only cases where I found that it does work and not for plastics is oil in an underground system, and you actually feed it in fertilizers and ozone to make sure there's enough oxygen so that it's actually energetically feasible for those bacteria to biodegrade it. That's why oil is so persistent. They don't want to do that. So that's my answer for that particular part, which I have some expertise in. It's really hard.
I can kind of speak on the second part because the reason I got into plastic science anyway was I did community outreach and citizen science stuff. So I think it's really important to engage with the community, and I did that a lot through Chelsea's U of T trash team. And I want to make sure I'm answering the question properly, but they asked whether or not how we can get people involved. Is that correct?
Yes, how to have citizen science engagement with
Yeah, I think citizen science engagement comes from understanding of microplastic in the environment. I feel like every day I'm very surprised about how many people don't know what a microplastic is. So I think it just starts at the beginning. We're educating people, and before we can get people to actively participate in that citizen science data. And there's a lot for macro as well. We have a lot of cleanup organizations that work on producing that citizen science data for people.
I just want to add, microplastics is challenging in terms of like, it's very challenging lab methods and stuff. Macroplastics is a great starting place. There's lots of like apps on your phone for when you do trash cleanups that you can record what kind of trash, what kind of plastics you're picking up, and that data is very useful. And maybe it's not as sexy as microplastics, but I think it's also like just as important. There's also a lot of projects right now thinking about dryers, and so I talked about it earlier, but Suzanne is piloting one too, and I've heard about these in other places, so I don't know, keep your ear out, because maybe there's a research group looking for volunteers to collect your dryer lint or whatnot. Speaking of which, if you know any laundromats in the Bay Area that want to collaborate.
I'll just piggyback on what Ezra was saying, that there's an organization, Break Free from Plastic, that runs an annual brand audit of its macroplastics. It's not microplastics, but it is a plastic waste in the environment audit, and you can do that anywhere in the world. And so it's a great way to get involved and you know, to contribute to the science. Wynne Cowger published a very well-received paper on that a year or two ago, using longitudinal results, and those audits are continuing. And there are many other organizations, and there are various apps available and things like that. But if you want to plug in, that's a great way to do it.
Okay, so I'm actively doing an outreach project right now. So to address the second question, there is, many of us probably know there's disconnects between the scientific community and the general public and policymakers. And I do believe that we should all be trying to make an effort to try and bridge those gaps in ways that you know, on a personal level, we feel comfortable doing. And so one way that I've been trying to do that for microplastic science, which seems like this very complicated, all this spectroscopy and tiny particles and all these chemicals, is by starting off with a community activity that the community can engage in, which in this case is beach cleanups, because we're actively removing plastic. And that's something that both people who would spend their noses in books, spend all their days sticking their noses in books, but also people who don't really know the science on behind it can engage in a way that helps with this problem, and then start to build those connections. So that's how I start, and then I use that to talk to people in the community, and some people they love picking up the trash, and that's all they want to do, and that's great. And some people get really fired up and they want to do more, and that's where we can start and work with them to do more and find ways to get them involved in our projects. So I think that's really important. Another thing that I think is really important for getting the community involved is scientists giving more presentations to the community. We give presentations at conferences and symposia like these, which are great, but they're not often very publicly accessible. And so having more platforms for scientists to give a talk, in science communication, but like give talks about what you do to people in ways that are not too filled with jargon, so that we can actually spread our message and get people really fired up and interested in this topic. And that's how we can then start bridging some of those other gaps in making change. I know I was talking to Neil a little bit about public pressure and these on an individual level, even if you only get a few people interested, it might not seem impactful, but you can really start to build up public pressure by getting people interested in the science you're doing.
And I just want to build real quick. One point is I feel like we also, just because we all study it so aggressively, we think it's very complicated, but it's really not too complicated. We teach 5th graders about microplastic. So I feel like if we take a step back and we realize that everyone else can understand this just as well as we can, then I think that really helps with bridging the gaps between people too.
Great points have already been said, but I just wanted to add that there are about a half dozen citizen science geared protocols for doing microplastics analysis, ranging from like 12th grade through like general public, undergraduate. There's also a number of education toolkits that are available, like the Wayfinders Society, or Wayfinders program from Algalita. Node, if I could recommend one for my citizen science, it would be Globe Nederland. They just published a really comprehensive toolkit for sampling microplastics that's applicable to lots of ages, with like lots of videos and lessons plans, and they'll even like send you toolkits of like the methods that you need to, or the supplies that you need. So yeah, it's out there, definitely.
Because everyone's talking about the citizens. Anyone here ever serve on a science panel for EPA or? I'm not talking about granting agencies. I'm talking about actually decision-making for a Superfund site. I know many of us have done that. And it is communicating with people who are not necessarily experts. Because a lot of them are people who are actually going to make the decisions. And I find a lot of what we end up doing is teaching. You know, why is this important? Why is that important? Why that's not going to work? Because people often come in, and I can say this especially now, into federal agencies with little knowledge or background or in thinking about using evidence to make decisions. And I have been on panels ever since I became an academic, for all these different agencies and so forth, through multiple Republican and Democratic presidents and administrations. And I find that's sometimes the most rewarding part is when you have people in a room and they can ask you questions. Why is this? And it's definitely not teaching, and I don't mean in a negative sense, because they're good at what they do, but they're not used to this. And it's a lot of work. And then they ask you to write something. And I'm not writing it for you guys. I'm writing it for someone, a different audience. I find that enormously challenging, but it seems to be helpful. The last thing I did something like that was the effect of war on the environment. If you want to be depressed, talk about that for half a day. But it was important to get some of the ideas across to the people about how this actually works. Why was I asked? I used to work for the Department of Defense. And it's just, I think that's one of the best things I feel I impact decision-making in a more direct fashion doing those sorts of things than, and I've done this too, talk to elementary school kids. It's just because you're talking to people who have to make the decisions. And during COVID, I did some of this, and it was the worst committee I've ever been on because it was very partisan. This was during Trump's administration, and it was so hard. And then some other scientists had come up with some crazy, I mean, you could definitely tell why they were on the committee, but it's like it was a couple of days of just hard work trying to communicate this to a broader audience. And I think that's something that we can strive to do. It's very constraining because to do most of this, you have to be a special government employee, which sounds special, but no, you come under, you're actually a federal employee. You're going, oh, well, you don't get paid as well, but also you come all under all the legal and ethical constraints. So it's a little different, but I would really recommend this. And this is why we have graduate students to train you guys eventually to do this. So I have a slightly different point of view. But I'm talking to, I know people in here have done exactly that same thing. Yeah.
Okay, we have time for one more quick question, and then we have to wrap up because we're already running over time.
I think that last question is a great lead into what I want to say, which is less of a question and more of a thank you. Something that we have spent quite a bit of time acknowledging just now is that when we want to have our work acknowledged and understood by the public, unfortunately, the onus falls on us as the scientists to communicate this to them. And this often feels like a Sisyphean task. Specifically in the realm of microplastics, you spend 90% of your energy picking, and I don't know, the rest, 10% of your time having nightmares about contamination, and often have few or less energy left than desirable to express why your research and your science matters. We don't go into this field because we want to receive many accolades. We don't go into this field because we want to earn a lot of money, or I guess we would be working for Exponent. Hi, my name is Fran DeCastro. My affiliation is UC Davis, the Bodega Marine Lab, Toxicology Lab. And so I wanted to extend a special thank you to all of you, especially in this time now when we're seeing budget cuts and is our work going to continue to be funded? And when we have to pick and choose between projects that we think might get funded or not, even though it's all equally worthy of being studied and understood. Special shout out and thanks to Dr. Tongri for your work with Gaia and also with international policy. As we all know at this point, microplastics are ubiquitous, not just through ocean currents, but also the grasshopper effect and global distillation. And what happens here has an impact everywhere. So thank you for all of the work that you do. I really appreciate you taking the time and the effort to talk at this symposium. And with that, I yield my time.
I just want to respond real quickly to part of that, this conversation about science communication, to say that there are people who are trained in science communication. Like, that's their whole shtick, that's their expertise, that's their job, and we should use them. Because scientists, we're often like awkward nerds who don't know how to communicate well, which is fine. We're doing this important work. Science communicators are trained to talk to scientists and then translate that. And so we should use that more. From a scientist perspective, like talking to the science communicators, that includes journalists. From the public's perspective, like being willing to listen to science communicators, even though they're not the one who did the actual work necessarily. And also art can really help with science communication, but that's like a whole nother thing.
Totally agree. I just want to quickly wrap up by thanking you all so much. I don't know about everyone else here, but I was a little worried about 7 full hours of talks and staying awake and focused, and really they breezed by because I learned so much. You all are great science communicators, and thank you so, so much for being here today. A big round of applause for all our speakers.
Final picture of the group and us.
Oh yes, and maybe we'll all take a quick picture if you have time.